Hello. Okay, welcome back. Good morning, everyone. Thank you very much for being with us today. This is our second day in the Biophysics 2022. Well, welcome back. Thank you very much for your attendance. And now, just a brief reminder of our sponsor institutions. This event is organized by the Universidad Autónoma de la Ciudad de México, UACM, Instituto de Ciencias Físicas de la UNAM, and Universidad Autónoma Metropolitana Lerma, which is where we are now in this beautiful building. Also, we are sponsored by Sociedad Mexicana de Física, Mexican Physical Society, particularly the Division of Statistical Physics and Thermodynamics, which I am the president of this division in the SMF. So thank you very much. We're now going to continue. I'm going to present you, I'm going to introduce you to Professor Carlos Muñoz Garay, which is from Instituto de Ciencias Físicas de la UNAM. He couldn't be yesterday with us, but now he is conducting the session. Thank you very much, Carlos.
Thank you very much for the presentation. Welcome. Then to continue this day, now is presented Dr. Irlanda from Sonora. Then please take the time for the presentation.
Thank you, and good morning, everyone. So thanks for attending this presentation. I would like, I would start my presentation and announcing that this this talk is like how the lectins can be used in biomedical application, and I will talk about the lectin that are working with in our lab. So here, I briefly introduce glycoproteins and their functionality. And so we can understand the importance and the role of the lectins. So glycoproteins are proteins that have linked carbohydrate or sugar to the structure. And for example, we can hear three different structures. And this structure, I'm sorry for my English, I'm very nervous. And the sugar tree had formed by oligosaccharide, one type of oligosaccharide, or can be formed by different type of oligosaccharide. For example, here we can see mannose. For example, this is a sugar tree of mannose. Normally, there are proteins that have attached glycan structure, commonly referred as sugar tree, and formed by a single type of sugar. For example, we can see here oligomannose, formed by unities of mannose, and other set of sugar tree carbohydrate can be found by hybrid or complex. All these structures have a core. This is the core for all this structure, and this core have two unities of N-acetyl-glucosamine, and the difference of this type, the sugar tree, is just here. So these proteins can have different, for example, branching patterns. So and when we can find these glycoproteins in the cells, many glycoproteins can be found in the cell membrane, just like this. where they function in cell signaling and cell adhesion. And many secreted proteins are also glycosylate. But what do these glycoproteins do? So glycoprotein allow cells to seek to hitch together, other, for example, and they also they allow cells to transmit information. But how can do they do so? So this is because their diversity. So we can find many different glycoproteins because all the different sugar monomers that exist. So there are different monosaccharides can be joined together, and there are many configurations of linkages, and the branching that is possible. So there are a high diversity of glycoproteins because there are different sugar monomers, and there are different configuration linkages or branching. So the extent of the branching or the site or position where the branching that's occur. So many more different oligosaccharides can be generated from 4 or 5 different monomic sugar that those oligopeptides formed by 5 amino acids. But why the complexity of glycoproteins? Nothing is ever tan complex in the cell for no reason. So one particular function of glycoproteins is interact specifically with lectins. Lectins are proteins or glycoproteins that bind reversibly to glycan or glycoproteins or glycolipid and polysaccharides. We can find lectins in all the different kingdoms of life. For example, plants have lectins, bacteria have lectins, virus, animals, fungi have lectins. And lectins are expressed on the surface of the cells or can be secreted to the cytosol. So if lectins are expressed in the surface of one cell, while glycoproteins are expressed on another cell, lectin glycoprotein interaction now how of those two cells interact. And that interact is very, very tight and specific. So the cell tag with other cell using a lectin glycoreceptor system. So because the cell, the cell send their message using glycoprotein, and the lectins can decipher this message encrypted of the glycoproteins. We can find different oligoforms of lectin. There are monomeric lectin, dimeric, tetrameric, and multimeric lectin. Each subunity have a recognition, carbohydrate recognition site. For example, this is a multimeric lectin from a snake, and we can see that each subunity have a carbohydrate. In this case, this lectin recognizes galactose, and we can see ligand interaction. So the interaction between carbohydrate and amino acid of lectin is mediated by weak interaction, and most lectin needs a metal cofactor to interact with the carbohydrate. In this case, this green balloon represents a calcium. It's necessary for this lectin. Because the lectin have one site for binding glycoproteins, they have ability to agglutinate cells. So there are many glycoproteins on the surface of red blood cells. For example, we know that different blood types found in the human population, A, O, B, AB. And these cells express different structure of this glycan. So the difference between blood types is related with the glycan that this cell expresses. For example, we can see this sugar tree expressed by blood types O is different to the B, AB, etc. So when the lectins recognize and bind glycan on the surface of the cell broth, red blood cell, for example, they form a net of cell and agglutinate. So this is an example of hemagglutination assay, because this way is common because we usually use this way to evaluate and determine the biological activity of lectins. And we use a microplate here, and we can see this positive hemagglutination, and we can see negative hemagglutination. For example, here the erythrocyte is precipitated in the wall. Also, there are many classifications of the lectins. According to their cellular localization, we can find extracellular, intracellular, cytoplasmatic lectin, membrane-bound lectin, and the same goes for the taxonomic origin. Also, according to their carbohydrate recognition, we can find lectin that recognize simple carbohydrate, for example, glucose, mannose, D-galactose, and we can find there are other lectins that recognize carbohydrates complex. Most of the lectins reported recognize simple carbohydrate. Also, according to their function, we can find hemagglutinins, adenosine, toxin, galectin, and selectin. For example, hemagglutinins are lectins found in the virus. Adenosine is in bacterial, and toxin we can find toxin in plants. Galectin, selectin in human or animals, for example. According to their structure, we can find calcium-dependent lectins, events, legume lectins, plant lectins with ricin B. These lectins are toxic, but they are toxic by this domain, so ricin B, and other lectins. With respect to biological activity, the lectin can have insecticidal, antitumoral, and antimicrobial activity. We can see some lectins that have been reported with antimicrobial effect. There are lectins with antibacterial activity. This lectin can form a canalopore on the cell membrane and inhibition and cause inhibition of bacterial cell division. And other lectins can recognize glycoproteins on the surface of virus, for example. Influenza virus have hemagglutinins that recognize glycoproteins on the surface of our cells. And this is the first step for the infection of the virus. With respect to antimicrobial and antiviral activities, we know that bacterial or virus express different type of glycoconjugate, and there are some lectins that can recognize this structure and that cause damage in the cells. But how lectins can be used in biomedical application, specifically in cancer? So the most cancer markers using in cancer are glycoproteins. So this is because the tumoral cells express different carbohydrates or glycan on the surface cell, in the surface compared with normal cells. For example, we can see the expression of the sugar tree expressed by normal cells and the cancer cell. And we can see the difference mainly in the expression of sialic acid and fucosa. So change in glycosylation, I believe it to be a main feature on oncogenic transformation. And we can use lectin to detect these changes of the cells using techniques, for example, immunofluorescence, microarrays. And this technique use multiple lectins and to detect change of the glycosylation and the one sample, for example, one experiment. So it's very fast and sensitive. Also, there are glycoproteins used as a clinical biomarkers of cancer now. For example, alpha fetoprotein and CA15-3 are glycoproteins used to track glycan chains in hepatocellular carcinoma and breast cancer. So also, we can use lectin histochemistry for a study of glycosylation chains in the cancer tissue. For example, this is a figure, in this figure we can see sample from a healthy woman. And here we can see sample from a sick woman. This is the breast cancer. And negative interaction is represented here. We can see a blue color. And the positive interaction is a brown color. So we can see that lectin staining shows the direct correlation of malignancies with intensity of staining. Also, the lectin can be immobilized and support, and we can purify glycoproteins of interest or markers of interest. Lectins can be antitumoral activity. This is because the lectin binds to, in this case, tumoral cancer, and they can cause inhibition of the proliferation in these cells. The lectins can be used in the design of drug delivery system because since a set of cell surface proteins are glycosylated, they can operate as a lectin binding. So lectin can interact with these glycan expressed in the cells, and it acts as carriers of drugs specifically to desired cells or tissue, for example. So we are working with legumes from Sonora Desert. Sonora Desert is a North American desert that covers the northwest Mexican state of Sonora, Baja California Sur, as well has Arizona and California in the United States. So we are working with lectins from 3 legumes, Vinorama, Palo Verde, and Palo Fierro. So we find that lectins from Sonora Desert legumes are tetrameric protein composed of subunities with identical mass. And the presence of metal ion is not essential to this to lectin carbohydrate interaction for this lectin. And they are specific to complex carbohydrates of glycoproteins like afetwin. This is important because the most lectin reported are specific to simple carbohydrate. We explore the stability, the thermal stability of this lectin using circular dichroids and agglutinin size, for example. And in particular, I show the stability of one lectin. This is for PF2, is a lectin from palo fierro. And we can see the PF2 display relative thermal stability and the native condition. And the melting temperature for this lectin is 72 Celsius degrees. Also, this lectin is stable to different temperatures. For example, we can see at 75 Celsius degree, PF2 have agglutinin activity too. Also, we evaluate effect of pH and the structural and activity of PF2, but this lectin is not very stable at different pH. It's important. This is important because if you want to use this lectin in biomedical application, we need to know their stability. For example, for can use in liposome or another system. This is analysis from amino acid sequence of two lectins from the cell legumes, PF2 and BL. And we compare their sequence with the lectins from common beans, PHA and PHL. And we can see that this lectin have high identity in their sequence. And also, this lectin, the recognition site is identical in all these lectins. But however, the specificity of PF2 and PHA is different. For example, PHA recognizes biantenary structure, and PH2 recognizes a triantenary structure. There are something important here, and it's just here. This is sialic acid. This is important because if we have a lectin that recognizes its complex structure with a sialic acid terminal, because the tumoral cell express high levels of this sugar. So, and this sugar, sialic acid, is related with malignancies and proliferation of these cells. So, so we decided to explore if PF2 recognizes tumoral cells, and this is the confocal image from this experiment. This corresponds to TPH1, this monomeric monocyte from leukemia, and these are tumoral cell lines from cancer breast. And we can see that PF2 binds strongly to the tumoral cell membrane. That means that this glycan is abundant in these cells. Also, we explore if PF2 induces apoptosis or necrosis in the cell. The cell, well, there are two ways to the cell die. One is necrosis, another apoptosis, and we can determine this way using a specific marker, for example, annexina or propuro ioduro. And we can found that PF2 exhibits apoptotic effects. For example, this is one of these histograms obtainable flux cytometry, and this is cell population, for example. And this histogram have 4 quadrants. Q1 represent apoptotic cells, Q2 represent necrotic cells, and Q4 represent viable cells. So we can see in the control, for example, here are viable cells, and we can see that the cells were incubated with PF2. there are increments in Q1 that correspond apoptotic cells. Also, we evaluate mitochondrial membrane potential. We use rhodamine 123. It's a cationic fluorescent dye that uses to specifically label respired mitochondria. And we can see, well, this is a control. We can see the rhodamine are present in the cells, but when the cells are incubated with different concentrations of lectins, we can see that it's not present here. So there are a lot mitochondrial membrane potential. Also, we observed if PF2 induced morphological change in the cells. This is example. These are the control of the cancer cells, breast cancer. And we can see, the difference is very evident. For example, we can see the cells incubated with lectin, and they are very ugly compared with the control. So for this reason, we decide explore if PF2 can change mechanical properties of these cells. Because it's known that the cancer cell deformability is correlated with increase in metastatic potential. So the metastasis required the penetration of cancer cells through narrow space, which is mediated by physical properties of the cell as well as their interaction with the confined environment. This is a complex process, but we can study, or we can do studies of mechanical characteristics of a single cell using a microfluidic chip, for example. Microchip bring new technology to study the dynamic of cells. So because metastatic cells have softer cell membranes, has deformation ability and microfluidic constriction canal. So we decided to study this using our cells incubated with lectin. So we are working with this microfluidic chip. So we are working with two designs. We can see the first design have constriction canal above the 8 micrometers. This is very, very, very small, because we need to know how occurs with the cells when pass for this constriction. So we pass of the cell for this, and we evaluate the velocity profiles and the elongation index, for example. This is a video. We can, sorry. ¿Puedes poner el video? We can see the cell. This is very, very fast, but we can see the cell moving on the constriction canal, stop here, and then continues for the way. So we are working with that, and apology, but we haven't finished the videos because there are so much videos to analyze it, but we are working with that right now. So in conclusion, there's the specific binding, the complex carbon with acid sialic acid, and grid stability observed for legum lectin from Sonora set could be used to for targeting drugs or tumoral T-cells, for example, because, for example, PF2 binds strongly to tumoral cells. Also, lectin can be easy isolated from lectins. It's very easy. In high quantities. And next there, antitumoral effect, showing they may become promised candidates for development of biomedical application. We have a open question, for example, what are the differences of the quaternary structure liver between PF2 and PHA? We need to do an experiment For example, we need to obtain the crystallographic structure of this lectin. Is the first step. And I would like to do simulation for this interaction too. Also, do the strong interaction with tumoral cells. Can Sonora cell legumes lectin be used on target de Villers agent? And what are the chains of the molecular and physics levels of the tumor cell exposed to lectin? This is important because there are works with this, with the natural compounds, for example. This is my our work group. They are my students. She is working with microfluidic. She is working with liposome using lectin, and she is working in identification of cell markers related with glycan. And he's working with future structural of lectins. And the last, I want to invite you this event. It's in December. It's a free event. And this event will be online also. And this is a picture from Sonora State. Thank you for your patience.
Thank you very much for the presentation. Excellent presentation. Everyone has one question? We are in time, please.
Very nice presentation. When you say that this branching of carbohydrates are expressed in a surface of a protein, you mentioned that this information comes from the genome, or is an enzymatic reaction that is attaching all these different monomeric glycans. What's the process in this?
There are a process. This is for post-traductional modification, the glycosylation. The glycosylation is a post-traductional modification that occurs in proteins in the cells, for example. There are other concepts used as like a glycation. It's different to a glycosylation. But the glycosylation occurs in the cells, and it's done by enzymes.
It's by enzymes, so the enzyme can recognize the surface of the cell when it's a cancer cell or it's a normal cell. So it's the enzymatic process that is recognizing all these differences.
The recognition is for the lectins. The lectins can recognize this glycan structure specifically, but glycosylation is the process. Used for the cell to adding carbohydrates to the proteins.
Okay, thank you.
Well, thank you very much for your nice presentation. I will start with the most naive question, which is, so you have said that there are many lectins, right? What do they have in common? Like, what do they have in common? All those lectins that you're saying, yeah, but what do they have in common? What is the chemical structure that they have in common?
The common structures of the structure.
Yeah.
There are, all the lectins have, all the lectins are similar at structural level. They are formed by, I don't know how to say, I'm sorry.
Yeah.
Can you speak in Spanish, please?
Era preguntarlo de que cuál era lo que tienen en común, pero es lo que dijiste al principio de las azúcares, ¿no? O sea, ¿qué es lo que tienen en común las lectinas, todas estas lectinas?
Oh, So all the lectins are the desert lectins.
Lectins, en
All the
general.
all the lectins can recognize carbohydrate.
So pueden ser tan diferentes, pero lo que tienen en común es que reconocen.
But the primary sequence is very identical.
Okay, okay.
So
And that sequence that is identical, do they have like, I don't know, like hydrophobic, hydrophilic interactions or?
yes. Mhm, there are.
Okay. And so how do the immunological system will react to those things? So you're saying that those can identify tumor cells, but what about the immunological system of somebody? Will they go against that and eliminate it? Or?
Okay. The lectins are not protein related with immunological, for example. There are antibodies that can recognize carbohydrate, but there are different to a lectin. This is because the lectins can bind carbohydrate the reversible form, and the antibody it's different, the interaction. But there are some lectins can have cause a reaction in the immune system and some bodies, for example.
And the last one. So I actually don't know if you say it, but with what you said at the end, it makes me think like, so those sonoran variants of lectins, do you have the structure of those? So you can make simulations out of them?
Yes, I would like to do that.
So but you have the structure? Okay. Well,
Yes.
thank you very much.
More questions? No? We are in time, no problem. Okay. And then I take time opportunity to present one small gift. Certified. Thank you so much. Okay. Continue? Because we have 20 minutos para preguntas, okay. Bueno, and then the next presentation is of the Dr. Jaime Ruiz from Universidad de San Luis Potosí. Welcome to the meeting. You can start.
Thank you very much to the organizers for inviting me. It's very nice to be here. We are going to talk about viruses, several viruses. Just let me understand. Okay. Since this is a biophysics meeting, I would like to give some examples of what has been done before, because nowadays biologists has done a great work in telling us how the biological system, especially in our system, works. But if we need to understand better, we need to go to the molecular level or atomic level as well. And these are some examples of what has been done in the past is the development of the structure of DNA, who was done by a group by especially James Watson and Francis Crick. James Watson was a biologist, Francis Crick was a physicist, and one name that was omitted there was Rosalind Franklin, who was a key person in the understanding of the structure of DNA. Another example is the study of the bacteriophages. This group, which is called the phage group, also have diverse persons working in this group, from physicists, chemists, biologists, etc. And I think if we want to develop science to understand better, especially in the range of biology and medicine, we need to get together multidisciplinary groups to understand better how things work. Oops, sorry. For instance, Richard Smalley said, human health has always been determined on the nanometer scale. This is where the structure and properties of the machines of life work in every one of the cells, in every living thing. The practical impact of nanoscience on human health will be huge. So it is important then to work together with physicists, chemists, biologists, so we can develop more science in this direction. So let's talk about viruses now. And the question is, what is a virus? And one of the main questions is a virus a living thing or is not? And the answer is that viruses are not living things. Okay? I will explain. How are they different from living organisms? Well, they are very small. Most viruses go from 18 to 350 nanometers in size. So, and although there are a few viruses that have been discovered that almost his size is about 1 micron. So, because of the size, we can see them using transmission electron microscopy or atomic force microscopy. We need this kind of techniques in order to see them. And they need cells to reproduce themselves. So that's the reason why they are not considered as living things, because they cannot reproduce by themselves. They need to get into a cell in order to reproduce. So biochemically speaking, they have either RNA or DNA, but they don't have both. They may or may not contain a lipid membrane. If they contain a lipid membrane, they are called enveloped viruses. If they are not contain the lipid membrane, they are called naked viruses. Viruses can have proteins inserted into the membrane lipid or directly attached to the capsid, and they are called glycoproteins, or since the COVID, the name of spike has been very used. So physically speaking, they are colloidal nanoparticles, which have the ability to interact with proteins or receptors located in the cell membrane. And this is one of the questions about the global warming, because people is concerned or worried about viruses that could be trapped in the poles, and because of the global warming, they can be liberated. And because viruses will, let's say, live there for millions of years, okay? Or stay there for millions of years, and they can be liberated, and because of the global warming, and people is concerned about that. So, biophysical or biology system, plant viruses, generally, they have different shapes. Here are the rod-like viruses, like tobacco mosaic virus, or the CCMV, which is like a soccer ball. Normally, these viruses that infect plants, they appear on the leaves of the plants. But also there are viruses that infect the flowers. And you can see the difference in the tulips flowers, that also, just as a comment, it induced a war in Europe in the 1600s because of these tulip flowers were very nice. And the price rise of the tulip flowers, and there was a war in, I think, in Central Europe. There are also viruses that infect bacteria, and they are very nice viruses, and generally the genome is pressurized inside the capsid, normally between 40 to 50 atmospheres of pressure. And they do not get into the bacteria, they inject their genome into the bacteria, and this was very important because the phage group used these kind of viruses, and they developed what is known as the modern molecular biology from these studies of the bacteriophages. And also there are viruses that infect animals like us. And we had some examples here. And normally these viruses have what is called the capsid. This is called the capsid and is made of proteins. And normally this kind of viruses has a lipid membrane. So they are envelope viruses. And they had these spikes or glycoproteins to recognize the receptors in the cell membrane that they will infect. You can see also the influenza virus is an envelope virus, and the smallpox virus that caused millions of people, of deaths in the past in human history. And this is the virus which is called vaccinia. I will talk a little bit more about them. And they are then envelope viruses. But here are adenoviruses. They don't have the lipid membrane, but they have the spikes directly attached to the virus capsid, okay? So, also, we have developed antiviral vaccines, and this was developed, or let's say, they started to be used in China about 3,000 years ago, when they controlled the smallpox scabs, and they add these scabs especially to children or people, and that gave some protection against the smallpox. And between the years 1300 and 1700, it is estimated that almost 50% of the people or the population in Europe have died of the viral agents like measles, yellow fevers, and smallpox. Around the 18th century, Edward Jenner in England introduced the concept of vaccination, and using a similar infection that was observed in cows, and he noticed that people who worked in that got infection, but it was not a mortal infection. And when the smallpox infection arrived, these people didn't get infected from the smallpox. So, what is the difference of these scabs that contains the cows, but also horses contain this kind of infection? So we had the smallpox, and we had the vaccinia virus, which is scientifically, their name is viralized vaccinae, and they belong to the same family, okay? And the vaccinia virus from cows was contained in the scabs from the cows. So this was the vaccine against the smallpox, basically. But people at that time didn't know how vaccines work, but they didn't even know that there was this kind of virus at that time. But they used these scabs from the cows to let's say, vaccinate people. So, from the development of viral vaccines, there has been many were developed at different times, but they didn't know how they work. But it wasn't until 1975 that we understand how vaccines work. And it was from the virus of the hepatitis B. What is it that we understood? This is an electron transmission microscope image from the hepatitis B virus. And you can see in this image that there are rounded circles in the middle of the image. That is the complete virus. It contains the capsid, the genome, and also contains a lipid membrane with the spikes of the virus. However, you can see that there are more particles, some elongated rod-like particles, or smaller rounded particles that didn't contain the capsid and the genome of the virus. And Baruch-Blomberg separated the viral particles from these particles, and he used these liposomes, basically, that contains the spikes of the virus as a vaccine. And that's how we understood how the vaccines work. Because our immune system recognizes only the external part of the virus, and the most external part of the virus is the glycoproteins that are there. So from that time, we started to understand how vaccines work. And that's the reason why now with the COVID pandemic that we have recently, we could develop vaccines very quickly. So then what is COVID? If you see the reports of COVID, people or the reports doesn't tell us exactly what is the size of the COVID virus. And you can see from this image that apparently there is not a homogeneous size on the virus. But my guess, because I had seen several images, is that as well as the hepatitis B virus, also when it gets out of the cells, it takes a lot of the cell membrane. And then you can see here, to me, this is the complete virus, and this is just a liposome, which is going to be cut here, and then it will have a liposome there with the capsid of the virus, okay? And there are some also images that people had drawn to try to explain how this virus is formed. You can see here the genome. The yellow part is the capsid, and the green part here is the lipid, the lipid membrane around the capsid. and we had the glycoproteins around it. So now, how can we make antiviral vaccines? Well, in the past, we used the same virus with weakened or attenuated vaccines, and that was used in the past where normally by physical or chemical methods, we debilitated the virus. So we injected that to the people, and it worked. But the problem with that is that if the virus is not attenuated properly, it could induce the infection in vaccinated people. So there was a risk. But nowadays, we know that what is important in vaccines is that the vaccine can develop inside the cells the glycoproteins. So nowadays, we can develop that vaccines based on DNA or RNA that codes for the glycoprotein, and it can be replicated inside the cells, and that will induce the response from our immune system. And here are some examples of the vaccines against the COVID. For instance, the Pfizer and Moderna just take the part of the genome that codes for the glycoproteins or spike proteins, and then they use lipid to encapsulate the part of the RNA that goes for the glycoprotein, and then it goes into the cells and start producing the antibody reaction from our immune system. And then if you get infected, you are protected against the virus. However, I took this image from the page of some of the pharmaceutics that use or produce the Pfizer and Moderna, and this image here is not correct, and I will explain it. Oh, can we return? Normally, for this kind of encapsulation of the RNA, they need to have positively charged lipids. And the positively charged lipids are cytotoxic. And I think that this is some of the reason why people, or some people, had very strong reactions against the vaccine, because they had to use positively charged lipids in order to interact with the RNA that produce the glycoprotein. And also, the way they put the image is not correct. And you can see that it's complicated to encapsulate with lipids the genomes. For instance, these are experiments that were done many years ago with DNA. And you can see that DNA interact with lipids and is not a very well-defined liposome, a spherical liposome with the DNA inside there. And if you use RNA, it's even more complicated, because DNA has a rod-like shape, very well-defined shape, while RNA does not. This is a secondary structure of the RNA. So if you put RNA interacting with lipids, it's not going to be very nice shape as well. I had looked for, in the literature, if someone has taken images of the liposomes with the RNA for the COVID vaccine, but I haven't seen any in the literature. And this is the Sputnik vaccine. They use also a viral vector as a transporter for the RNA that codes for the glycoproteins of the virus. And they use another kind of virus. They use an adenovirus. From the virus, they took the gene that codes for the glycoprotein and they put it inside this capsid. Okay? And then, once it is in that capsid, it is used as a vaccine. And they use another adenovirus and to make a reinforcement of the vaccine. But they are using what is called virus-like particles as vaccine. Okay? And the vaccine, also the AstraZeneca Oxford, use a different kind of adenovirus, which infects chimpanzees. And as a vaccine, they took off the genome of the virus and they put inside the genome of the RNA that codes for the glycoprotein, and then it is used as a vaccine as well. So if you get vaccinated, now you are protected against the virus. If you have any questions, you can interrupt me anytime. In general, then, there are vaccines that use lipids as a vector to take the RNA into cells, or there are more vaccines based on virus-like particles. Okay? And there is also a vaccines that only use the spikes. They join the spikes and form a particle, or something similar to a particle, and it is used as a vaccine as well. And here, I did this table some time ago when the COVID was very strong. But you can see that most of the technologies used for the production of vaccines are based on virus-like particles. And I think the virus-like particles are more effective as vaccines than using lipids or liposomes. There are several vaccines, and at this time, all these vaccines has been approved, basically here in Mexico. Now we work with the cowpea chlorotic mottle virus, which is a virus that infects plants. It is a plant that is like a bean. And normally, this virus has a size of about 28 nanometers, and the capsid is made of 180 copies of the same type of protein. And one of the things is that the proteins have positive charges towards the center of the capsid. So they had about 10 positive charges, and that is important because the proteins attach to the genome, which has negative charge, and then start forming the capsid around the genome. And you can see here the shape, which is rounded, a rounded shape. But as I mentioned before, there are viruses that infect plants, viruses that infect animals, and viruses that infect bacteria. And it is supposed that viruses that infect plants do not infect animals or bacteria. Viruses that infect bacteria do not infect plants or animals. And viruses that infect animals do not infect bacteria or plants. And one of the reasons that is believed is that, for instance, a virus that infects plants cannot enter animal cells or bacteria, and the way around for the other type of viruses. Okay. Here is a phase diagram that we elaborated some time ago. And in this region, which is here, located at about an ionic strength of about 1 and a pH of about 7 to 7.4, in that region, we disassemble the virus. And using some techniques, we can separate the genome from the capsid proteins. And we purify the capsid proteins, and then we reassemble only the capsid proteins without genome at different pH and ionic strength. And in this region, we obtain capsids, empty capsids, with a similar shape as the virus. However, it was surprising that in other conditions, we can obtain multicapsid shells or tubular shells as well from this protein capsid. And here are some of the images. This corresponds, for instance, this corresponds to this region, which has similar shape as the normal virus. But these capsids are empty. They don't contain the genome. And these are the multicapsid shells that are located in this region. And one of the things that we discover is that the phase diagram is also associated with the electrophoretic mobility of the capsid and the protein. Here is the virion, the blue line, and when the electrophoretic mobility is 0, yes? Yeah, yeah.
And that is, so you told us that each copy of the capsid has roughly 10 positive charges.
Yes.
And those are important to talk to the genetic material of the virus.
Mm-hmm.
In these empty shells, all those positive tails are looking into the capsid, right?
Inside the capsid.
So who takes care of them? Who neutralizes those charges?
They are not neutralized specifically, but the tail is small, and also we have the interactions, the capsid, lateral interactions between the capsids that form to form the capsid. Even though we have the positive charges, and one can think that they will interact to repel each other. But the interaction, lateral interaction is stronger in that sense.
Right, because you do not need a high ionic strength
No,
to make those.
In fact, we can have here the capsid form, empty capsids form at low ionic strength. Yeah.
That's nice. Thanks.
I was telling you that the electrophoretic mobility is associated with the phase diagram. And here, the blue line corresponds to the virion, and when the electrophoretic mobility is 0, it is associated with the start of this zone where we have separated the empty capsid from the multicapsid formation. And here is the electrophoretic mobility, the red line, of the protein only. And it is associated with the end of the formation of the multicapsids as well. And you can see here images of the multicapsid formation. And here we have the sum of the tubes that we observe. Even though this virus is rounded, the empty capsid can form tubes as well in these conditions of pH and ionic strength. Coming back to the interaction and the formation of the virus, normally we have the capsid. When this is empty, we have the positive charge inside, and normally the capsid outside is negatively charged, the proteins. And here is the RNA, and this is an RNA that codes for the green fluorescent protein, and we put this inside the capsid. And we can use this for gene therapy. And for instance, these are virus-like particles that do not contain the virus genome. They contain the genome that goes for the fluorescent protein. Okay? And these are the particles that we obtain. And for our surprise, we broke some of the biology paradigm because it is supposed that plant virus cannot enter animal cells. And as a test, we just put a cell culture from humans and put the solution with the viral particles that contain the gene that codes for the fluorescent protein. And for our surprise, the cells start fluorescing. It means that the capsid enters the cells and delivers the gene, and the cells start making the fluorescent protein. And that was surprising for us and for many people as well. It was, the next one? Here is the also more images of the fluorescence. And it took us a lot to publish this paper. And sometimes the editors just say, I don't believe in this. I don't believe that a capsid from a plant virus can enter directly to a human cell. Okay? But finally, we could, after we sent it to about 10 journals, we published it. But took us a lot of effort. And because we are breaking some of the, yes?
Another question. You showed us that the capsid is negative on the outside,
yeah.
right? So one would imagine that RNA would not want to lie at the surface of these
the
VLPs.
capsid. Yeah.
Still, that sounds like an obligate control. No? You have to make sure that all the RNA is inside.
Oh, yeah. We had the images that I showed you before. We didn't detect any RNA outside.
But would you, but that's microscopy, no?
Yeah.
Would you be able to see the RNA?
Oh,
like
yes.
that? Or I was thinking a control more along the lines of treatment with RNAs. So you have the VLPs already prepared with the RNA inside. You treat them with RNAs.
Yes.
That would make sure that there's no RNA floating around or on the surface.
No.
And then you use those.
No, because also we determine how much RNA we need to put depending on the concentration of the protein as well. So
I know, it sounds like very carefully done. I still, if I were the editor, I would have asked for that experiment.
Yeah, okay. But the thing is that, but also RNA cannot enter a cell directly if it is not inside the capsid. Mm-hmm. So they were the capsids that entered the cell. Mm-hmm. So also we were working on the COVID vaccine in my group, and when in 2020, and this was the idea that we had about the vaccine. In fact, we did have the RNA that codes for the glycoprotein of the COVID-19. And the idea was to interact it with the CCMB capsid protein and to make the virus-like particle with that, to get it into cells, to produce the glycoprotein, and then purify the glycoprotein, and then also with viral-like particles already with the genome that codes for the glycoprotein, we can also attach glycoproteins, and this will act as a double vaccine, because this glycoprotein already will act as a vaccine, but also these viral particles will contain the genome that goes for the glycoprotein. But this will take longer to be authorized as used as a vaccine. But another idea that we have is to use gold nanoparticles and to attach to the gold nanoparticles the glycoprotein. And because gold nanoparticles are already used in medicine for treatment, different treatments, this could be authorized more easily. However, our CONACYT approved only about 130 projects in that time, and only one project was on vaccines. The rest of the projects were statistical analysis and things like that. Okay? And I think that the project that was approved was the Patria vaccine in that time, which is already authorized here in Mexico. But we could have done also the vaccine, and we just got onto here. We made the viral-like particles with the genome, but we couldn't continue because of the lack of budget. However, we have the same idea using the capsid of the CCMV virus, and this is the virus of what is called the I forgot the name. In Spanish, it's the white spot syndrome virus. That is called. That infects shrimp. Okay, it is mortal to shrimp. They kill shrimps. This virus. Okay, and we developed a nanotransporter based on the CCMB capsid, where they form rods because the vaccine uses DNA. So the DNA is elongated, and we form the rod-like particles, and we use that as a vaccine, and it works. We give it to the shrimp, and then we make shrimps that were vaccinated, and we put them in contact with shrimps that were infected, and the shrimps vaccinated were not infected over time. And the shrimps that were infected, they die. So with the same idea of using the CCMB capsid, we did a vaccine already for this animal. And also, there are several works that are worried about how to get different medicines or for treatments into cells. They use viral nanoparticles, proteins, graphene, dendrimers, carbon nanotubes, fullerenes, or silica particles. And the question is how we can get this into cells or inside the cells. And what we had done is we prepare nanolipospheres of about 20 nanometers in diameter that contains anti-cancer medicine. However, 20 nanometers is a very small for this kind of liposomes. And they are unstable. And why? Because there is a physical process called Oswald ripening when this kind of nanolipospheres get close together, they attach to each other, and they fuse, and they grow in size because of that. And normally, the people that authorizes or the agencies that authorizes the application of this kind of treatment will not authorize something that is not stable, okay? And to make it stable, we put it inside the capsid of the CCMB, and the problem was resolved, okay? And you can see here, we measure the size of the nanoliposphere as a function of time, and you can see it increases with time. And then, inside the capsid, it is stable, okay? And also, we are doing the encapsulation of different gold particles that, for instance, the nanorods, gold nanorods are now used nowadays, used in phototherapy for cancer cells or cancer tumors. And also, there are some called nanoshells that are made of gold as well, and we encapsulate it and we stabilize it. Because one of the problems with the nanoshells is that in about 2 weeks, they disappear, they dissolve, okay? But encapsulated, they are more stable. Also, one of the problems with the gold nanorods is that normally during the synthesis, they are covered with a lipid bilayer, normally with positively charged lipid bilayers, okay? And that is cytotoxic to cells. And that's a problem because even if most of the nanorods goes where the tumor is located, if it goes to a normal cell, it may kill it as well because of the cytotoxicity, okay? What we did was to replace the positively charged lipids with negatively charged lipids, and then we encapsulated the nanorods as well. And we are working on that, and with the nanolipospheres, with the nanoshells and nanorods, we are working now in vitro experiments against cancer cells to see how effective they are. Another thing that we had done is to envelop the naked capsid of the CCMB by a lipid bilayer. You can see there that the capsid is enveloped, and the idea is we can put in the lipid any kind of receptor that recognizes specific cells, and then with the capsid inside that carries something specific for medical treatment, we can get it into the cells. And we are working on that. We are about to be submitted this publication. Also, one of the problems nowadays is the bacteria that are resistant to antibiotics. And one of the things that is used against this kind of bacteria are silver nanoparticles. But the problem with the silver nanoparticles is similar to the gold nanoshells that we work with, is that they disintegrate in about 2 weeks. They are not stable, the silver nanoparticles. Okay? But the problem with the bacterial resistant to antibiotics is a problem that is growing. But as one of the speakers yesterday talked about the bacteriophages, this was the group led by Max Delbrück, and they also recognized many years ago, about 100 years ago, that this kind of virus can kill bacteria. And they were proposed about 100 years ago to be used against bacteria. But the penicillin was discovered, and this was forgotten. Okay? However, this is an example of the epidemiologist Stephanie Strait that has her husband infected with bacteria resistant to antibiotics. In 2016, she contacted several researchers that worked with bacteriophages. And she asked them to donate bacteriophages that kill bacteria and made a cocktail of different bacteriophages and injected that to her husband, which is about to die. And it worked. Okay? And I think that the bacteriophages are some of the things that we can use against bacteria resistant to antibiotics. And how it works is that the bacteriophages injected the gene into the bacteria, and the bacteriophages start to replicate inside the bacteria, and the way they get out of the bacteria is by destroying the bacteria. They kill the bacteria, and this is one of the things that we can use against bacteria resistant to antibiotics. One of the things is that for millions of years, these bacteriophages and the bacteria has existed, and most bacteria, or I can say all bacteria, has the gene that codes for the bacteriophage inside their genome. Okay? And we need, but the genome of the bacteriophage is dormant. And we can activate it. And one of the possibilities to activate the bacteriophage inside the bacteria is by UV light. And we are working on that at this moment, trying to make different bacteria cultures and try to activate the bacteriophage inside the bacteria and purify the bacteriophages and make cocktails of different bacteriophages to apply to people who is in danger because of this bacteria resistant to antibiotics. In conclusions, the understanding of the structure, the physical interactions and properties of viruses provide great challenges for physicists, chemists, and biologists, both experimental and theoretical. And the good thing is that we are making groups which are multidisciplinary, and I think that will help to develop this kind of research for treatments more quickly. So understanding the most important interactions, protein-protein, protein genome, capsid membrane, etc., one can extend the knowledge about the cell assembly of the virus and take advantage of that knowledge for the design of virus-like particles for medical application, such as vaccines or antibacterial developments. And this is my group, and this is a multidisciplinary group. We had physicists, we had chemists, we had people in material science, and molecular biologists in my group. And also, we had collaborators in my university and also at UCLA, Chuck Nobler and William Gelbart. Thank you for your attention.
Thank you for the presentation. Thank you. Someone have any question?
Hi, thank you for really nice presentation. I have 3 questions. So the first one is a curiosity. Those vaccines that work with positive charge lipids, is that related that for the reason to have them in extremely low temperatures to keep them like functional?
You mean because
like the
they have,
Pfizer,
but the positive charge, the positively charged lipids.
Yeah.
Yeah, they can work also.
Yeah, my point is like if those positive charge lipids are the reasons why they need to have them on extreme low temperature.
No, not necessarily.
Okay.
They have the positively charged lipids in order to interact with the genome that they will carry to the cells. So they can involve the cells, the genome, with the electrostatic interactions.
Okay. The second one would be like, so you have pretty much proof that vegetal capsids can interact with animal cells.
Mm-hmm.
So would that mean that is the genomic information the one that does not infect animal cells? Because then we can get in contact with them, then you have proof that those things enter into our cells, but we don't get anything.
Yeah.
So what's the reason?
Yeah, that was a question that we had for a long time, because when we discovered that the capsid or the virus that infect plants can enter our cells, we asked ourselves, why then these kind of viruses do not infect us? And the answer was given by a collaborator that works in biology, and she explained to me that it is important because the reproductive machinery inside the cell will take the ends of the genome, in this case the RNA, had to take the ends, but the ends has to have some particular structure. And apparently the virus that infect plants do not have that structure, so our reproductive machinery inside our cells cannot reproduce the information that is in the virus genome of this plant genome.
Yeah, that's really interesting.
But people who likes vegetables, if they eat some leaves, they have some decolored leaves, it may be that they are infected by some plant virus.
And that would also imply that in the future there may be a mutation where it can be transmitted to us.
It is possible.
Yeah.
It is possible in the future. Yeah.
Yeah.
But also, for instance, I didn't mention it, but at this moment, I can tell you that on your skin, you had a lot of bacteriophages, because bacteria that we had in the skin may have been killed by bacteriophages that remain in our skin. We had a lot. Yeah.
Interesting. Well, and the last one is you show us a really nice phase diagram of those capsids forms. So I don't know if I miss it, but have you tried like the efficiency of those different forms entering animal cells?
No, we had only the virus-like particles that are rounded, similar to the virus, And also recently, the development of tubular ones that also work. The multicapsid, we have not worked with that yet.
Okay, so there may be that you may find something which is better, like interacting with.
Yeah.
Okay.
Yeah.
Thank you very much.
More question?
Thank you very much, Jaime, for your very nice talk
Thank
and
you.
for this broad introduction on viruses. My question is that, well, you mentioned most of the properties, I would say, about viruses, but you almost not mentioned about the structure of the genome inside the capsids. Also, you mentioned yesterday that they have this coil-like structure and also a toroidal structure. But taking advantage of your last slide, where you mentioned your collaborators, I have here a paper of Nobler and Gelbart. And they mention that there is a very well-known structure of the capsid, this icosahedral, for instance, in adenoviruses. But there is a very little known about the structure inside for the genome.
Yeah.
Can you explain us what is the problem there? I mean, why the experimental techniques have not developed yet to understand exactly how the structure there?
Yeah, there have been experiments for DNA, in the case of DNA, because some of the viruses, or the bacteriophages especially, they have some small molecules with positive charge inside the capsid, and when the DNA gets in, they interact and with the negatively charged DNA, so they form the toroidal shape inside the capsid. But this is for DNA viruses. And not all of them has these molecules with positively charged molecules, but some of them has that. But in the case of RNA, it's more difficult. And as far as I know, there has not been experiments, for instance, with the RNA, working with the positively charged tails of the capsid that gets inside. These positively charged get neutralized most of the negatively charged RNA charges, but not all of them. In the case of the CCMB, for instance, it has 180 proteins, and each protein has a tail with 10 positively charged tail. So it will be 1,800 positive charges inside the capsid. But normally the RNA has about 3,000 negative charge, okay? So there is still repulsive interactions, and I didn't mention it, but in most images of the CCMB virus, the center seems to be empty in the images. And my guess is that the RNA is ordered inside the capsid, and because of the repulsive interactions, the center gets basically empty in there. But I have not seen experiments on that.
We are on time. Some question? No? Okay. And then, thank you so much
Thank
for
you.
the excellent presentation. And take opportunity to a little gift for you. Now it's time for the break coffee time. All people is welcome to the next step. Two steps. So, we can continue working, talking about this interesting topic, biophysical meeting.
our session of this morning. It is my pleasure to introduce you to Professor Tom Chow from UCLA. He's going to talk about a stochastic model for transcription translational coupling. Thank you for being here, Professor.
Thank you for inviting me. Yes. Hello? Hello? Is this good? Yeah, okay. All right, thank you for having me. So today I want to talk to you about a specific project we worked on in the stochastic theory of ribosome RNA-P interactions. And this is in the field of what's called translation-transcription coupling. This is when the translation and translation are somehow correlated. And so let me just give you a very basic overview and sort of set up the problem. So as you know, of course, there's the central dogma, DNA, RNA to polypeptide, right, to proteins. So it turns out that two very structurally different scenarios for this to happen. One is in prokaryotes and one is in eukaryotes. In prokaryotes, well, the transcription is both made by RNA-P, RNA polymerase, but in the prokaryotes, they make the polypeptide, the mRNA that's made by the RNA-P can simultaneously be translated by ribosome. Whereas in the eukaryote, there's a lot more complicated processes going on, and the mRNA needs to be transported out of the nucleus. And then there's other signaling factors that initiate the translation by ribosome. So we'll focus on the prokaryotes today, which is bacteria, which is where most of the experiments have been done. And it's a simpler process, a simpler problem, fewer parts, if you will. But you'll see that both of these things can be happening simultaneously. So you'll be producing mRNA, but also producing protein at different numbers. And there's kind of an overlap because some of the mRNA is not completed yet, but the protein is already being translated from it. Okay, so this is, in general, what they mean by what I call TTC, translation, transcription, coupling, or transcription-translation coupling. Okay, so that's the setup. And so here's a picture of what's going on. So you have the DNA that's in a bacteria, a nucleoid, let's say. And there's some initiation of RNA-P, it forms a bubble, and it moves forward. And as it's moving forward, it's reading the codon, the DNA, and then adding the right nucleotide to produce the mRNA. So as it's moving along, of course, it's producing more mRNA. When it reaches the end, maybe it falls off or continues growing, but this gets cut, and that's your complete mRNA, okay? So you can see this forms what's called a TASEP, a totally asymmetric exclusion process. So it is a process where it's a one-dimensional lattice. Think of it as a one-dimensional lattice. You have particles on each site or group of sites, and they can move forward. This is the transcription elongation rate. Let's call it Q. Okay, so it's moving forward. It waits for the right nucleotide to come in, and then it moves forward, adds it to the mRNA, and moves forward with probability per unit time Q dt, right? A rate. But then if they're blocking each other, then the one behind one cannot move. So this is a traffic jam model, so to speak, on a lattice, discrete. And then when it gets to the end, it falls off. Okay, so you've got this chain. You have feeding in, right? And then you have exit. And then so there will be a current of these particles. So there will be a current of mRNA being produced. Number of complete mRNA produced per unit time is the number of RNAPs crossing this termination point per unit time. Okay. But we also know that RNAP, I mean, there are some experiments. This is a relatively simpler problem where they have done single molecule experiments. So we know that if the wrong nucleotide gets loaded, right, and it doesn't match, so there will be some pausing. That's one purported mechanism for pausing. Others are just internal states of the RNAP, right? So as it's moving forward, it can pause, and these are single molecule experiments, which you can read more about. There are many of them, actually, from this group in Germany. So they can track the nucleotides transcribed as its transcription, and you'll see there'll be, instead of just so this is the end, so it's moving forward with some velocity, but you'll see at certain junctions, it will stall. Okay, so the RNA is moving, and I change state, or I load the wrong nucleotide, and I just wait here for a while until it's fixed, right, until I do some error code, let's say, correction, proofreading, and I fix it. So during this period, the RNAP is stopped, but it's not because it's blocked by an RNAP in front of it. It just stops spontaneously. Sometimes this is also called backtracking, because when it stops, it's very loose. It stops and it diffuses back and forth locally. Okay, so there are these pauses. Okay, so that will be important in our model. Okay, so here's a picture. The wrong nucleotide. It's paused, or it's called paused or backtracked. It's sort of the same kind of name. So it can't move forward, but the others still can, of course. Okay. So things move forward, and then the error is fixed, let's say. Right, the wrong nucleotide kicked out, or some other more complicated molecular process, which I won't get into. Then it can continue moving, right? So every time you have a pause, you have a pileup behind it. Okay, so that's one picture. But then also, which I haven't pictured here, the very first picture I showed you that there's also ribosomes now that can access this mRNA. So the ribosomes will bind to the mRNA and also move forward and add the right amino acids every time to form the polypeptide. So this is happening simultaneously. So this is a picture of a completed mRNA. There's, let's say, some rate of initiating the ribosome, and as it's moving along, it's adding the right polypeptide until it gets to the end, falls off, and you have your protein. Okay? So this also is a TACEP if you look at it, right? So assume that the mRNA is completely detached. It's a lattice. It's a one-dimensional lattice. I add ribosomes and I move forward and then produce my protein. And the same thing, it moves forward with some rate P, but then it's blocked if there's another ribosome in front of it, okay? So I think this is another TACEP, another one-dimensional, totally asymmetric exclusion process. So in fact, so here's the picture of what's happening. So this is what I'll be looking at. So you have the RNAP moving along a gene. There's a start end, a termination end. It's producing the mRNA, which is here in this purple line. These particles are the ribosomes that attach with rate alpha and move along the mRNA, all right, producing the protein. So this is a schematic of the full process put together. Okay. Okay, any questions? No? Okay. So what happens when of a single TACEP? So you can see from this picture, there's this 1D lattice here, and then each of these is producing an additional one-dimensional lattice. These one-dimensional lattices are actually growing, right? So the lattice size is actually increasing until it gets to the end, then it's completed. So what happens, just as a reminder, what happens with one TACEP, okay? So this is a well-known result, and there's many follow-up works on it since the mid-90s or so. So for an isolated one-dimensional TACEP, we can calculate the steady state current, right? That's an injection at one site and exit on the other. What is the total flow of particles per unit time in the steady state. It turns out you can represent it in terms of a phase diagram, which I'll just remind you of here. So I'm going to plot the entry rate, alpha, and the exit rate, beta. And the current falls into these three classes for an infinite-sized system, right? Take the hydrodynamic limit. P is the forward hopping rate. Let me make sure. So Q for this, P for this. So I'm referring to this lattice, right? But P or Q is the same thing. It's the forward hopping rate. Initiation rate alpha, exit rate beta. Okay. So what happens is, and then this is the size of the particle d, right? So if I just have one particle per lattice site, then d is 1. But so as you can guess, my unit size will be 1 codon, right? 3 nucleotides. The RNA P and the ribosomes are bigger than 3 nucleotides, right? They're bigger than 1 codon. So D will be 10 or something. Okay, so what happens is, let's say, so the phase falls into 3 current values, right? If the entry rate is high, and the exit rate is low, we're in this regime. So the exit rate is low. That means the off-ramp at the end is slow. That means I've piled up, right? There's a traffic jam. And the current will only depend on the exit rate, right? Because that's the rate limiting step, is someone to leave. Similarly with the other case, with low entry rate, high exit rate, the only time I get a current is when a particle comes in to my highway, right, to move through. So this is low density, high density, and in between, when both are high, then you have what's called the maximum current phase. In this region, the density is about half, if you will, right? There's equal number of holes as particles. That gives you the most mobility, right? You have enough particles to transport, but you have enough spaces for them to move, right? So that's the maximum current regime. So this is very interesting. So if you think about this, now we have 2 of these, right? We have 2 nested T-steps because you have the T-step for producing mRNA, the T-step for producing protein. So if you just do a very simple, rough estimate, so this is the mRNA concentration at steady state, let's say, okay? So forget about the T-step, let's just look at the very end. How many RNAPs are flowing through per unit time, making complete mRNAs? That's the production of mRNA. Then mRNA has degradation as well, so I call it mu mRNA. So at steady state, you would expect the mRNA number to be the current over the death rate, right? The production rate divided by the death rate. The protein concentration at steady state would be the number of mRNAs times the ribosome current along the mRNA, right? I have many mRNAs. Each of them has a ribosome current on it. So I multiply the ribosome current by the number of mRNAs divided by the protein degradation rate. So the total protein concentration, right, just substitute that into there, is J times J. J RNAP times J ribosome. It's a product of the 2 fluxes of the nested T-step. So this is interesting if you think, and so each of these currents can be in one of these 3 regimes, right? So in fact, that should be a 9, but okay, I know why I put a 6 there. So there's 3 currents for each of these. So there could be 9, right? So you get a very rich phase diagram, even at steady state, for 9 regimes of protein levels, okay? And why I put 6 is here because typically for the RNAP, it just moves past the termination end a little bit. There's no beta, right? The beta is the same as the Q, so it's not so relevant. But conceptually, you can have 9 different phases. So this is kind of like the picture of mRNA and protein production, in some sense uncoupled, right? I'm talking about completed mRNA and completed protein. Okay, so, and this is the steady state flux. So, but what are the experiments, right? So that's steady state, and that's just the level, right? But what are more interesting, so I'll get a little deeper now, are these experiments that measure mRNA and the associated protein. Okay, and there's some kind of assay that measures the protein. This is remember, this is the square root of the protein level, and you may be able to see why here. Okay, so this is steady state. So what if I wanted to do the time dependence of this problem? Okay, this coefficient will still be there, but then there'll be a function of time multiplying this, right? Sorry, back this way. So these experiments are on different bacteria, different conditions, and of course, some of them are related to the LAC-Z operon, different conditions, lactose-rich or glucose-rich. And what they measure is, at time 0, you start the process, I guess it's maybe with GTP or something, you allow the first RNAP to bind and transcribe. Okay, that's time equals 0. You turn the system on. And then you have to wait some amount of time, some seconds, before you start observing the level. Okay, so this is still increasing, right? And so if I waited long enough, this would go up and then saturate at that level. That's because then the degradation would come in, right? That's the previous plot where I had J over the mu. This is just the very initial transient now, right? I'm not looking at steady state, just looking at the initial transient, and it's linear, roughly, okay? Also, so this is the problem, right? So mRNA abundance after induction, it's the current times the amount of time that I've been producing mRNA, right? So d-protein dt is this, the amount of mRNA times the current, the protein current, ribosome current on each mRNA. So it's this, so you would get something like this. So there's still this product times the t squared. This is for short times, right? At long times, this function, as I said, will flatten, right, saturate because of death, of degradation. Okay, so now we look at the start. So these experiments, right, are actually bulk experiments. So this is a whole cell culture, right? So it's averaged, right? So it's not one gene at a time, or it's not the result from one cell, which of course will be a stochastic process. So this is the average, and then this is the average of the protein production. Okay, this is square root, so it's the squared. It's because of this issue, right? You first start producing mRNA, then you have a very few mRNAs, and they're starting producing protein. But then, as the mRNA increases, then you increase furthermore your protein production. So it's quadratically increasing, whereas this is linear, right? It's because this is producing that. So if this is linear, this is quadratic. Okay, so they took the square root, and you extrapolate, and you try to find when did you first see it? When did you first see the protein after you first see mRNA? So many cases, it's roughly about the same time. But in other cases, there's a big delay. Okay? So something is going on there, right? There's a delay. And this is what I want to focus on in the rest of the talk. Okay. Okay, so those are the experiments. So I'm not going to look at the full nested Tay-set problem. That's a more computationally difficult problem, right? You have many of these, of the mRNA Tay-sets, and then on each one you have to have a Tay-set. So you can imagine the state space is really large, when you have large numbers of mRNA. Okay. I mean, let's say for simulation purposes, it would be very difficult. So all I want to do is now just focus on this part, right? So I'm going to collapse the problem into something much simpler by just looking at the leading ribosome for each RNAP, or we're going to average over them, right? In some sense, we're going to just look at one case, right? So assume you're looking at the very first mRNA, this long one, and the very first ribosome, right? Let's just assume that. So now that's a single molecule system, if you will, right? And I can analyze that. So in here. Okay, what else is important in this model? All right, so there's this delay, so why did people study this, right? I didn't really give you the biological background, maybe. There's many processes like termination by Rho proteins, okay? So the Rho protein will come in right after the RNAP and stop the process, okay? So that's another level of control that the cell might have. Other reasons are, this could be a very short-lived process. The mRNA could have a high degradation rate depending on the condition, right? So if that's the case, then as you first start producing mRNA, you want the ribosome to go on it right away. Where if it doesn't, and you wait too long, this site could get degraded, and then you wasted, producing that mRNA, right? So there are timing and control reasons for wanting to correlate or do this in concert, the mRNA production and the protein production, okay? And especially here. Okay, then, so we're going to sort of now mathematize the problem, right? Put it in sort of a statistical physics model picture. Okay, so here we're going to assume a gene of length L, that's L codons for that gene, 300, 400. Alpha, I'm going to use as the initiation rate for the ribosome. I'm going to assume that t equals 0, the RNAP is already here, right? At t equals 0, it's already initiated, okay? And then these will move forward. So the ribosomes move forward with rate p. The RNAP moves forward with rate q. There is a association constant between the leading ribosome and the RNAP. Right? Think of it as binding. Ka association, Kd dissociation, binding and unbinding. This is also known to be mediated by proteins, like they're called NUS-G and E or something. So there are other proteins that, so there is some association with this, some weak binding, but there's also proteins that can bind to the RNAP and then stick to the ribosome, okay? So that's effectively having the ribosome bind to the RNAP, right? That protein will be in the background. I'll assume it's in the background. So all of that will contribute to Ka and Kd. Then there's a distance from the starting site for the ribosome, which I will call M, and then there's a distance from the start site to the RNAP, which I call N. Okay, so the two discrete variables M and N now. And this is a case where it's tightly coupled, right? It's touching the RNAP, this leading one, for example, here. Okay, and the experiments now, what are they measuring? They're measuring the T ribosome minus the tRNAP. So T ribosome is the time for the first ribosome to finish. Okay? So that has to finish after the RNAP finishes. So this is greater than that, and so this is my delta T. So this is what they were measuring here. This is delta T. Okay? So what are the quantities? Activation first at t equals 0. So then we have to define a few other quantities, the average RNA-P and ribosome velocity when they're free. All right, assume this RNA-P, forget about the right, just assume one system, right? So it's just a random walk. Well, biased random walk, only moving forward. What's the time it takes to get to the end divided by the time it took? So that would be the average velocity. Okay, and then we have the average velocity of the ribosome as well on a completed mRNA. That's just the average. These are decoupled quantities, right? So we're just defining these quantities to get everything clear. So now I want to ask, what about interactions, right? How will these quantities change? Okay, so let's summarize the variables. And I mentioned stalling, backtracking, right? We have to put that in. So this model has a few parts, but I think we can keep it clear. So you have the ribosome initiation, ribosome hopping, RNA-P hopping. K plus and K minus will be RNA-P stalling and unstalling rate. Okay, so K plus, I think I've defined it as being stalled to moving again. Okay, and then K plus star, so this is also known and has been modeled as well. So if I'm stalled, then I'm backtracking, I'm sort of fluctuating around. If there's a particle behind me, it can, in some sense, push. The push is really entropic. All right? There's a particle behind me, I'm not allowed to backtrack too much anymore. Then I will correct my error or jump over my error and go forward. Okay? So this you expect to be greater than K plus. So K plus star is the rate of going from the stalled state to the processing state again if there's a particle behind me. All right? And this is just with there's no particle behind me. So now when you have this like in this case, well, not here, but let's say this were right here touching this, and this was stalled, right? Then this would go into the unstalled state faster, right? It's getting a kick from behind to move forward. Okay, so now you think, okay, maybe the ribosomes doing this, letting it push. Yeah.
Sorry, I have a question. For instance, this K plus star wouldn't actually depend on the number of particles that is behind the other one because it's just one particle?
Just one particle, yeah.
Just one particle?
No, what behind is only just one particle.
But it could be more than one particle, maybe.
But they're way behind. So it's only this one. It's only this one here will not affect this. It's just this one preventing it from sliding back, right? So, but that's a good point. I'm looking at just this one, right? But there's another RNA-P back here. So this RNA-P can be kicking this one forward, or it could be this ribosome kicking this one forward.
Actually, I was thinking that.
Oh, you were thinking that. I see. Yeah, right. I'm only focusing on the leading part. The other part would be the full nested Tay-Seps that I introduced in the beginning. Then it gets very, you have many different steps, and that simulation is very difficult. And I don't see an analytic way yet of approximating it. Okay, so we have this. Now, right, M is the ribosome position from this part. N is the RNA-P position from this start. Then L is the total gene length. Then there's also, I'll put two other parameters, which I hope we can keep clear. One will be an interaction range, L. That is the range at which you can use Ka and Kd. That's the range at which they combined. So they have to be close, right? A few nucleotides. I mean, this part is floppy, right? This is single-stranded mRNA. So once they get close enough, here's the ribosome, here's the RNA-P, then I can bind, right? But if they're very far, then I don't have Ka or Kd, right? This one cannot have Ka or Kd on this one, right? Only this one or this one, very close. So this is a length L. If M and N, sorry, if N minus M, right? N minus M is less than L, then I will allow this process in my stochastic, the whole process, right? Okay, this LP typically could be larger than the L. That is what I'll call a protection distance. That means if this is within LP of this, then my rho protein that I mentioned cannot get in, right? Rho protein is size LP. So in order for the rho to come and stop to kill this process, I need to squeeze in, right? So this distance has to be, so if this distance is less than LP, then I cannot terminate with rho protein, for example. Okay, so these are the main parameters. Okay, now, so from that we can already just sort of say something, right? So what is the effective velocity of the RNAP? So the RNAP moves forward with rate q. Okay, but then I said sometimes it stalls. When it stalls, it doesn't move. Then I have to unstall to move forward again. So the average velocity, if you will, including assuming stalling and unstalling is fast, or assuming an infinite, very long lattice, and I allow many stalling and unstallings, the effective velocity is just this q weighted by the time that it's in the plus state. Right? If it's in the minus state, in the non-moving state, it's 0, it doesn't move. Okay, so it's just q times k plus over k plus plus k minus. So that's the effective velocity of the RNAP. Now, I have p, the velocity of the ribosome. So what if the velocity of the ribosome, what if the velocity of the ribosome p is much greater than q? Not the effective q, not q bar, but just q. Then p is always jammed up against this, right? It's like the driver that's tailgating you, always pushing you from behind, right? Always, okay. But if p is less than q, and the driver in front, so the driver in front is faster than you, and he doesn't stall, then you never catch up, right? It'll just run away. It's called runaway in this process. So in that limit, it's runaway. So what we'll focus on then is the interesting regime, which is q is greater than p. That means if the RNAP did not stall, it would run away. But it does stall. So, but if it stalls, its speed is q bar. Now what if p is comparable to q bar, or a little greater than q bar, let's say? Yeah. Yes, yes, yes. About twice as fast. I have a chart of numbers, yeah. 3 times maybe. I use 12 and 30 codons. The structure of the what? Yes, the expressome. That's within L. That's what I will describe. I don't use that terminology because it's very confusing to me.
Oh, sorry.
They use expressome they use collided expressome and interacting expressome.
So the thing is that the interaction between the RNA polymerase and the ribosome is so tight when they are connected via a messenger RNA that it has been isolated and the structure is known. It is deposited in the PDB.
Yeah. So what I'll calculate is the likelihood of having an expressome, and that's and how we define it in this model is the little l, if it's within the L, or LP, because some of the expressomes are mediated by the NUS protein, so that's bigger. So I put in those 2 parameters just for that purpose.
Thank you.
Okay, so yes, as you said, the Q is actually larger than the P, right, 2 or 3 times. But it's not necessarily much larger, but P is not necessarily smaller than Q bar, including the stalling, right? You have a fast driver, but they get stopped by stoplights. Then you have time to catch up. Now, when you catch up, you can, the NUS-G or direct expressome, you can bind. There's some cases, so there are some experiments that show the strong coupling and some that don't. I think it depends on condition and bacterial type and maybe even the gene. All right, so then that's your redemption. This car is driving very fast, but they get stopped by a stop sign. You can catch up, you bind, and then something happens, right? Then what will happen? So we will explore that. Okay, so this, oops. Okay, so we're here. So then it's in this case where what I call TTC can emerge, transcription, translation, coupling, right? Otherwise, it would just run away. You can never catch it. Okay, so this already sets our parameter regimes. Now the model, the way I've described it, reduces to a very simple, discrete lattice type of a model, right? A discrete stochastic model. So this is M, and this is N, right? This is the coordinate of the ribosome, and this is the coordinate of the RNAP. And the RNAP has to be greater than or equal to, I start at 1 here, has to be greater than or equal to the coordinate of the ribosome, right? Because it's in front. Or it could be right at each other, which is here. So then, so at t equals 0, I start with the RNAP already at the initiation site, here, right? And then 0 means no ribosome yet. So then you remember there was a binding rate of the ribosome to the first site. So I start the RNAP right here at the first site. It starts moving. But then there's a rate of the ribosome coming in. So there's a little bit of delay, 1 over alpha roughly, right? So I start the ribosome, RNAP, it's moving up. And then at some point, the first ribosome comes in, right? At position 1, I go from 0 to 1. 0 is no ribosome, position 1, and that's with rate alpha. Okay, so now I start here. So this is a random walk, two-dimensional random walk on this boundary, in this state, starting right there. And the starting position depends on the ribosome initiation rate. Okay, so you can see the story in this picture, I think. This is the L, and let's say this is LP. Let's say it's bigger, right? So there's a zone of states where N minus M is within L. That's in here. So these states, the two can bind and unbind, of course. These states are free. So let's say I start my random walk here. The hopping rates are Q and P, right? So it's tracing out a random walk until I get to until the RNAP finishes. And then the ribosome finishes. So this is my delta T. Okay? Now, suppose I have a system where the ribosome is fast. Then it's this blue curve. Then I move along here, and I bounce along here, and I get here. And my delta T is very small. Right? And I stay in this gray region much longer. Right? So depending on the parameters, I have these trajectories, and we want to analyze the trajectories and make some claims. Okay, let's see, I didn't miss anything. Right, there's the protection zone. Okay, so here's the system. Here's the model then. I just said it all in words, right? Each site now, so this isn't just a MN state space. The state space is within each M and N, there are other internal states. Those internal states are stalled RNAP, for example. So I could be at a state MN, my RNAP is at site N, but it could be stalled or unstalled, right? So there's two states within that site. Okay? This is what we call the free region, where D is N minus M, the distance between them. different from the D from the TACEP that I described. That's the size of the particle. This is the distance between the two particles. Right? So there's a K plus and K minus. So in this state, I can go up or to the right, P or Q. But also, I can have this Q, I can have this state be paused or go back. Okay? So this is the free state. Now at the boundary, so in this gray region, remember I said that I can have binding, association, and dissociation, right? So these are our four states. If I'm right at the boundary, P can't go forward, right? Because D is 0 here, right? D is 0 along this line. They're abutted against each other. So I can't move forward. But this one can go into, the RNAP can go into the stalled state. Now, going back is going to be, because there's a particle behind it, I can go back faster, right? K plus star. Remember that? We discussed that, pushing from behind. Or, now that I'm close, I can associate or dissociate. If I associate, I draw it with these black little lines here. That means they're bound. Okay? So if they're bound, right, this still can't move forward. This still happens. I can go into the stalled state or unstalled state. However, I will let this RNAP move forward still. How does it move forward? Particles bound behind me. There's this rope. I move forward by pulling in, right? By moving forward, that RNAP, that extra mRNA goes in between the two. So there's like a pocket, right? Actually, it's called exit tunnel for the RNAP that can store 10 or so codons, right? So that I'm going to call that the same as L. So if I'm here, I can still, so maybe this is a picture, right? I can still draw in our mRNA, right, by moving forward. So now so this is a slightly different case. This is inside the interaction zone. I can still have association and dissociation. I can still have stalling and unstalling, right? But the unstalling rate is K plus, not K plus star, right? Because there's no particle right behind me. So I can unstall at a slower rate. I can bind, right? But even after binding, this can move forward by pulling in more particle, pulling in more mRNA and moving up. Okay. Finally, this case, at the very boundary, okay, at the very boundary, everything I said here can still happen. But now, the RNAP cannot move up in this micro state, in this interior state. Why? Because I've already drawn so much mRNA that I cannot cram any more between me and the particle behind me. All right, so I'm pulling in. I won't let that happen unless I dissociate, right? So I can dissociate and then I can move up. Okay? So now just sort of, put these pieces together and think what effectively, just, zoom back. These are little details. What is happening here, though? What does this gray region mean? I can enter it, right? I can move in and go in, but then when I want to leave up, I have to dissociate. So that's an extra delay. That's an extra step. So you can imagine then, this is what this is happening. This is exactly the expressome, the coupling that we'll talk about, at least framed in this sort of statistical physics way. So let's look at the states, internal states. This is L, the boundary. So these states here are the ones in the gray area, okay? So this is the distance. So now I'm projecting it into n minus m. So that's d, n minus m. So if this is less than or equal to L, I'm in this state. I come in and I go back and forth. This is A is associated. A equals 0 is bound. A equals 1 is bound. A equals 0 is unbound. A is for associated. This parameter B right? This B is for backtracking or not. Okay, so here, right? So B is 1 is backtracking. So I'm not backtracking, my D is 1, and I am bound. That's this case right here. Then I can go into the backtracking state and go back, okay? So these are all the configurations. So imagine this set is within that gray zone. I enter it, then I have these other states that I spend some time in, and then I have to go into the unbound state, A equals 0, before I can leave again, before I can leave. I cannot leave from here to here, right, to greater D. I have to go back to this and go in. So think of this as like a well, if you will. You come in here, there's more states, right? There's more entropy here. I've got to stay here longer before I can go back out. So this gray area is like a zone of attraction, if you will, really. It's like effectively, it's a attractive well. So, right, I'm doing this. If I never hit this, I do this. But if I hit this, I more prefer to stay inside here. Okay, so let's see. Okay, so these are typical parameters we found. Some of these had to be guessed because they're not directly experimentally observable. The pausing, unpausing rates, you know, a few tenths per second, K plus. So we're going to define the dissociation rate as the association rate times e to the minus EA. So there'll be an EA and an E plus that determines what the association-dissociation rate ratio are, and the E plus is sort of the acceleration from behind to going into the precessive state. It's the kick that the stalled driver behind you wants you to push you, make you go forward. How much of an effect they have is this E plus. L and LP, a few codons, right? Just sort of a molecular size. Okay, so, right. So what do we do with this? Okay, so the full system we can simulate with just like a Gillespie type algorithm, event-based algorithm. Okay, you saw the state space. It's not too big, right? It's this triangular state space, but there are other states inside each, right? So it takes a little time, but it's not too bad. These are the rates for the Gillespie algorithm. So I'm defining my variables now as my state space as M, N, A, and B. A and B describe whether it's associated or whether it's backtracking. Okay, so those are my 4 discrete parameters. And I can look at all the transitions between them, right? And where they apply, right? Some of them have to apply when D is inside L. For the KA, for example, right? I have to be within L. Okay? So you could just tabulate this and we can simulate. Here are some examples. L equals 4 and 40. So here, 4 is down here, so I'm sort of in that little well, right? I'm moving back and forth. Then I have a sort of an escape event. I escape, but then the RNAP pauses, and I catch up, and I go back here, right? So it's the intermittency of these trajectories that we want to sort of analyze, right? So the rest of the plots I'm going to use standard. These results change depending on the parameters, but we're going to use the best guess parameters here. Okay, it's not that important, right? But I just want to sort of describe the physical features. Okay, so that's one approach. Another approach, I can write the master equation. So the M and N is a 2-dimensional random walk. Each internal site has 4 possible states inside it, right? The A and the B. A equals 0 or 1, B equals 0 or 1, right? Associated or not, backtracking or not. Okay? So you can define the probability as now a vector. Okay? And it's the probability that I'm at. So I'm going to separate out the spatial coordinates, M and N, from the internal binding and backtracking coordinates, A and B. So there are 4 states of A and B for each m and n, each position that I'm in, right? So this becomes a vector master equation, if you will. And the transition matrices, right, these would be the hopping rates in the standard 1D problem, but because we have internal states, these are now matrices. The hopping rates, it's a hopping rate matrix. So this, we can find a numerical solution to this. And it's a little more involved, it turns out. Like so many things today, it's just easier to do the simulation rather than do the deeper path integral. You can think of this as a path integral along those state space paths, right? So one can do that, but that's actually more work. And it turns out to be even more computational time than just brute force simulation. Anyway, however, there's some cases where this is treatable, and that's the steady state limit. If I take the length of the gene to be infinite, and I just look at the steady state distribution of that one can do somewhat analytically. And hope I have some time to show. Much time? 5? Well, okay. Okay, so, so if we assume P and Q, okay, so the P and Q's can depend on the codon, the site that I'm at, right? So let's ignore that. Let's just assume it's constant. We can actually do some analytics. And this is the analytics, what I was just saying, assuming an infinite lattice and assuming steady state. And I want to look at the dwell times. These are the mean dwell times of being in D less than L versus D greater than L, right? It's basically the dwell time inside of here, below this line, L equals 4, or above it. And for this one, L equals 40. Below or above it, how much time it stays, the mean time it stays in each of those positional states. With that, one can define some of the things that we'll calculate, which now I need to get to quickly. Okay, so those experiments that I mentioned were bulk experiments. Now we're doing a single molecule type of stochastic model. So what other new metrics should we define, right? They only had delta T. That delta T was the mean delay between finishing the protein and finishing the mRNA. But since we have a full stochastic model, we can define the mean in a different way. We can define this as the expectation of these times. These times are now stochastic. If I take this delta T, that's the difference between the T ribosome and tRNAP, right? I can define the whole distribution of times instead of just the mean, right? I have the whole distribution. I can define a coupling coefficient. This is really sort of defining, this is how we define coupling, is that when things are finished, I am in the A equals 1 state, the bound state. So when I finish the protein, I'm bound to the RNAP, right? At the very end. That's one, so it's only the end state that I'm worried about here for the C, right? It's coupling coefficient between 0 and 1, right? It's a probability. Then another one is the fraction of time protected. So that is these trajectories. Let's say this one. I start here and I end up here, finish here. What is the fraction of time I stayed in the gray region, right? So that fraction of time protects me from a premature termination or row coming in or whatever, okay? So those are the, so we're able to define these quantities within our model, calculate those quantities, and in fact, approximate them with the analytic approximations as well, okay? So, so this is our result. This is the distribution, okay? Rho delta T, density of that time. And you can see there are a number of cases. If there is the pausing and then binding and unbinding, you actually get, you see this goes up here, right? Up towards 1. So it's really multiple peaks, right? So the experiments only measured the average of these distributions. With our full, more detailed model, you can get the whole distribution. And in fact, in some of these distributions, you can see multiple peaks, okay? So A equals 1, you see? This is a case where, so the solid lines, you see the solid lines? They go up. The solid lines are the cases where there's a lot of binding. There's a high association. So if you include binding rather than just exclusion, like in the TASA model, just being close, that's not enough. Here we're saying there's actually binding. If I'm binding, then I'm in that gray region. I spend some time there before I unbind again, right? I spend a lot of time in the coupled state. You can actually have multimodal densities, okay? And then this is the coupling coefficient, the mean protection time. These are our analytic approximations. I should probably, right, okay. So since I'm low on time, let's see. Let's see if there's something interesting I want to say. Okay, yeah. Let me just mention this then, and I'll finish. This case, right, is the most interesting. So the ribosome behind, we said, can kick a stalled RNAP to make it go forward if it's stalled, right? However, if that kicking amount is low, right, that the acceleration, right, the E+, remember that's how likely the driver behind you will kick you out of your stalled state. If that's low, we can actually show a decrease in the transcription velocity as a function of P. So P is the rate of ribosome moving forward. What I'm saying here is I can increase the rate of the ribosome moving forward, and I can slow down the whole thing. Okay? So why is that? It's because if I have too low of a rate, I'm rarely within that interaction zone. If I increase P, I let the ribosome come up close to me. It doesn't push me out of the stalled state, right? Suppose the case that the ribosome doesn't push me out of the stalled state, but it comes up close to me that I can bind, Ka, right? If I bind, then I'm in that little attractive well in the gray zone, right? So then I'm bound together. And if I'm bound together, remember, if the RNAP is bound to the ribosome and I move forward a bit, I can only draw a certain amount of mRNA in, and then I cannot go forward anymore. I have to unbind before I can continue moving. So that little delay causes a slowdown for a number of these cases, right? So I can actually have a faster ribosome slowing down the coupled process. And it's all due to entropy. It's all due to the different number of states possible in that gray zone. Then there's also a trade-off. We can also define the translation efficiency. That's the velocity of the ribosome over P, right? So this has to be less than 1. P is a free ribosome moving through. The velocity will get blocked because there's an RNAP in front. But if so I can plot this as a function of P. This is low P, increasing P. If I have very low hopping rate, then the efficiency is high. But the expected protection fraction of time is low, right? So the ribosome is very slow. So the efficiency is high, right? Because it's divided by P. Nothing impedes me, so it's high. But if I'm slow, then I never get within the interaction zone. I never get close to the RNAP to protect it. Right, so it goes down like that as I increase P. So that's a protection and efficiency trade-off, right, that we can define in this model. And anyway, there are a number of results. Protection time, so that was the average protection time, but we can calculate the standard deviation of it, get some more statistics from our simulation. Yeah, a number of other interesting things, but you can read about it. This is the signaling process, so anyway, this is describing the bimodal behavior. And here, this analysis shows that the bimodal behavior, it relies on the binding, but it's most sensitive to the bimodal behavior of the RNAP velocity, right? Because the RNAP can be processive or stalled. It's that process that generates the bimodality in the delta T. Okay? The other is monomodal. The bimodal part comes from the RNA-P. Okay, so just to summarize, we sort of developed a stochastic model for this set of measurements, usually under the nomenclature of translation, transcription, coupling, and then we really define what the coupling means in a more mathematical way. Define a number of parameters, L and LP, that describe protection. And also define new metrics that hopefully can be measured in single molecule experiments instead of just the bulk experiments. And this should appear soon in Biophysical Journal if you want to look at it. Okay, so that's the end for my talk. But I also wanted to mention so I'm from a UC, which has a program with you guys, I think, with maybe Connect, or I think, and it's so actually there's a lot of funding, and I think it's maybe not so utilized, particularly for undergrads that want to do a PhD or a graduate student who wants to do a postdoc at any UC. Right? I think it's like the funding is quite generous, but there are some strings attached. I think you have to come back to Mexico. You can't use it to immigrate. That's the thing. Yeah, but that's it.
Right.
But if you're interested, look here.
Maybe we have time for a quick question while you are ready.
Perhaps I miss it, but you are assuming you have like an infinite supply of ribosomes and RNAPs, right? So the abundances do not take part in the model.
This is just a single molecule, right? The taste set that I talked about, where the immigration rate is alpha, that alpha, of course, will depend on the concentration, and that's assumed fixed.
Okay.
For the taste set, for the multi-particle.
Yeah.
This is just 2 particles.
No, I was wondering about the coupled TASEPs, because I mean ribosomes and RNAPs are really big proteins and they are expensive, so it's not that you will have as much as you can, but that's considered in the alpha.
That has been considered by other. The phase diagram was just fixed alpha, right? But if you put in, if you have many genes competing with each other for the RNAP or the ribosome, then that's a conserved quantity that has been treated, in the mean field limit.
Okay, thanks.
The mean currents.
Thank you. Thank you for this very, very nice and interesting talk, Tom. So I have a question about the modeling. It seems to me, maybe I'm wrong, but it seems to me that you can model this as well as 2 random walks couple, and then one of the walkers of this random walk can, of course, can block the other one from moving forward, and this case are assuming the role of this P and Q probabilities in a one-dimensional random walk. Is that correct or not so?
So you mean, well, this is I just decomposed it into a walk in two dimensions.
Yeah.
But you're saying 2 one-dimensional random walks?
Yeah, 2 one-dimensional random walks when one is superimposed to the other one and can stick to the one, and I don't know, maybe.
But okay, that's where we started with.
Exactly. Oh, I see.
That is. Wait a minute. That is this model, right? Exactly. This distance and this distance, and then it's the way.
So you move to
to
another.
To the 2D representations.
Yeah. Thank you.
Let's thank again.
Thank you.
Thank you. The next talk is Nina Pastor. It is our pleasure always to have in this conference to Nina Pastor, our great professor from University of Morelos, UAM, Guaymor.
I think that we are older than the other YM. So the other one is the YMX, and we are YM. No. Well, we don't call ourselves like that. So thank you for having me. I know that this is the slot right before lunch, and everybody's desperate to go and get some fresh air. So let's try to make this lively and expedient. Please interrupt me at any time, no problem. And if we run out of time, I can tell you later, no problem, okay? So this is a collaborative project with Jordanis Perez-Llano and María del Rayo Sánchez Carvente. This is an experimentalist lab that works with fungi, specifically with extremophilic fungi. They look for fungus that can grow at high salt concentrations or that can grow in petroleum, things like that, because they're interested in biotechnology to try to then later recruit whatever the fungus makes in order to clean up the mess that we make. Okay? So they do the experiments, and in my lab, Marco and I do the computations. And so the systems that we are modeling with their particular sizes are dictated by what they do in the lab, not the other way around. Okay? So how do I make this run? Okay. No. The what? This one. Okay, here we go. So as you know, protein sequence dictates the structure of the protein and also its dynamics. Okay? And the structure and the dynamics can also be modulated by the environment. So when an organism is faced with a challenge, there are many options to cope with that challenge. So if you're thinking in the ecology level, then the answer is to live in a community. So whatever you cannot do, your neighbor will be able to do, and everybody's happy. If you're thinking at the physiological level, there are basically two options. Either you get rid of the offending challenge. So say you live in high salt concentrations, you pump the salt out, and that way you protect yourself. And the other option is that you live with it. And that means that you have to tailor the molecules that you make in order to withstand whatever the pressure is. Now, if you're going to go that way, then in your genome, you have to have a collection of paralogs. These are variations of a single gene that can be expressed at different conditions to meet the challenge at hand. Okay? Now, it's not enough to have the collection. You also have to express them at the right times and in the right concentrations, and that is a transcriptome. Okay? These RNAs are further, their translation into proteins is also regulated, and that leads to the proteome, okay, which is defined as the collection of different proteins and the relative amounts at particular times and places. And also, you can engage in post-translational modifications. So you already made the protein, and now you can stick different things to the amino acids, like phosphates or acetates. Okay? And that leads to a connection with a metabolome, which is the collection of metabolites that the cell has at a particular time. So if you want a really fast answer to an acute stress, what you do is that you use post-translational modifications to what you already have in your proteome. If the stress is constant, then it might be better to make new proteins, right? What I'm going to show you today has to do with the analysis of a collection of paralogs and orthologs. So we're going to be comparing a family of proteins that are expressed in a single organism, and also we're going to be comparing different organisms. Okay, So thinking about proteins, of course, when you stress a cell or an organism, all of its molecules get stressed, okay? But proteins are the engineers of the cells. So that's why we focus on them, and also it's because they are the polymers that we understand best. So you start studying where you're most comfortable, no? Anyways, when you place a protein in an extreme environment, that is something where that's not its native state conditions, then you have to take care of these three issues. One is that the protein has to be both chemically and structurally stable, okay? So in order to ensure structural stability, there are two options. One is to have more interactions between the amino acids of the protein, and that, say, you guarantee by increasing the number of methyl groups. You have a variant that doesn't have a valine, but has an isoleucine or a leucine, so you have more contacts between the amino acids. Or you can use small molecules that you have in the cytoplasm, and then you bind them, and that lowers the free energy of the native state. That's a good idea. The other option is to have a larger free energy of folding, not by stabilizing the native state, but by destabilizing the unfolded state. The net effect is the same. The free energy of folding is increased, and then you're more stable, okay? And the other is to alter the vibrational modes of the protein. So you change the heat capacity, okay? That is a strategy that is commonly used to deal with high temperatures. Now, regarding the second point, when you stabilize a protein and you get carried away, one of the things that can happen is that you make it so rigid that it no longer functions. So remember that proteins are not rocks. They are closer to a piece of jello. Okay? They have to move around in order to work. So you have to make sure that you have the adequate amplitude of motion and that these motions happen in the right time frame. Say you have an enzyme, it better breathe at the right time in order to make the chemistry good for life, right? And that has to do with the correct population of the excited states relative to the lowest native state that are good for the function at hand. And finally, it's this kinetic aspect of being able to respond quickly to drastic changes in the environment. And this is where post-translational modifications come in. About those, I'm not going to talk anymore. Okay? Actually, this talk is centered on the second point, which is the population of excited states. So, when one is trying to understand the link between dynamics and function, one has to choose a protein family to work with, because we have found that each protein family has its own recipe. Remember that living systems have a history. We are the reflection of many millions of years of tests. Actually, we have the recipes that have survived throughout this long time. Okay? All the individuals that made a wrong choice could not reproduce, and they are no longer represented in our gene pool. Okay? So anyway, given that each protein family has a different recipe, I cannot make broad generalizations about which is the right strategy to withstand high temperature or very low temperatures or high salt or metals or whatever you want to think about. So I have to choose one family and see what that family does. In order to look at protein families, then you can choose either orthologs, which are the same protein but in different organisms, or paralogs, which are the same protein, well, variants of the same protein in one organism. Okay? So in this story, the stress of interest is sodium chloride. I don't know if you're aware, but there are bugs that can grow in halides, which are these rocks that are basically crystalline sodium chloride with a little bit of water in between. So what can we expect about the mechanisms for salt adaptation? Regarding stability, what has been found is that proteins that belong to salt-loving organisms tend to be net negative in charge. So if those guys are placed in a medium that has very little salt, all these negative charges together with no counterions, that's an electrostatic bomb. So the protein literally goes kaboom, and there goes your native state. So you need enough salt to make the protein charges happy. Now, on the other hand, high salt could be destabilizing because salt gathers water around it, and then there's not enough water left to solvate the protein, so it falls out of solution. Okay, so normally high salt has two problems. There's an electrostatic contribution, and the other thing is that you're left without free water to solvate your protein. Now, regarding structure, when you have a lot of salt to go around, what is going to happen is that the side chains at the surface of the protein are going to be enticed to talk to the salt, to the cations and the anions in solution. And that could push the structure into a non-native structure. Okay. Regarding dynamics, again, at low salt, since you have a lot of repulsion between like charges, the proteins are going to be moving around like crazy. And that may be non-functional. Okay? So that's an excess of flexibility. Remember that we went the right amount. That's called the corresponding state hypothesis, put forth by Jennecke in the 90s. Okay? At the other end of the spectrum, if you have a lot of salt, then you freeze the protein. And it has been shown that if you, I don't know if you've seen this fish that is cooked with a layer of salt on top. So it makes like a crust when you cook them, when you cook it. And then, of course, in order to eat the fish, you have to get rid of the crust. Well, with high salt, with the proteins that have very charged surfaces, there is a layer of sodium ions that is forming a crust on top of it. So then the protein would like to move, but since the negative side chains are coordinating the sodium ions, you have no motion. Okay? So the good news is that we can look at all of these possibilities when we perform molecular dynamic simulations, because we have explicit treatment of the protein, the water, the sodium, and the chloride. Okay, so that's what we're going to try to do. Now, I warn you, this is work in progress because we found a bug last week. So we are repeating roughly 25% of our data set. So this is just a flavor of what we want. If you want the full story, invite me to the next version, and I'll tell you how it came out. Okay, so our favorite proteins are called hydrophobins. And they are produced by filamentous fungi only. No other creature so far on the face of Earth produces this family of proteins. So here what you have is your regular mushroom, and if you look at the structure, you see that there are air channels in the tissue of the fungus. The hydrophobins like to line these air channels. So their favorite place for living are the air-water interfaces. Okay? That's where they spontaneously assemble to form these structures that look like a collection of matches, small matches. They're called rodlets. And this is, I think it's AFM. This is an AFM picture. So this is really low resolution. We have absolutely no idea what the protein really looks like when it's like this. When they are in solution, the hydrophobins, when they are monomers, they are perfectly soluble in water. Okay? But if you place them near a surface or an air-water interface, then a miracle happens and they switch into rodlets. So that's the miracle we would like to understand. Now, why do the fungi want hydrophobins? So you have a spore in water and it germinates and it starts producing hyphae, and that's in water. But if you want to produce a mycelium that is in the air, you have to break the air-water interface, so there's the surface tension problem that you have to overcome. That's what hydrophobins are good for. Okay? Also, this is like a raincoat because it allows the budding structure not to lose that much water by exchange, by evaporation. Also, it facilitates attachment to the hosts. So when you have a fungus that's growing on a substrate, the hydrophobins help the attachment to the substrate, and that substrate could be your lung tissue. So there are many fungi that, of course, produce hydrophobins that cause very nasty diseases for us. And they also protect the fungus from our immune system. So it would be nice to understand them and how they form to see if we're interested in this line, if we could devise a small molecule that would prevent this hydrophobin layer formation. Okay, hydrophobins are small proteins. Luckily for us, 70 to 150 amino acids. They have a beta sheet core, always 4 beta sheets, okay, in this particular topology. And what you can see is that we have a hairpin, and then we have a crossover, and then another hairpin. Okay, and that happens in all of them. What makes them different is the amount of amino acids in the particular loops, and then the secondary structure that you can find there. These structures are stabilized by 4 disulfide bonds, and these are illustrated down here. And they have a very strange topology also. Okay? We are going to be looking at what happens between cysteines 3 and 4, between 2 and 5, and between 7 and 8. Okay? Because those are related to what happens in this loop, and in this loop. Okay? Whatever happens in the amino terminus, we have not looked at that yet, because that is in an intrinsically disordered region, and we are scared of those for the time being. So, I forgot to tell you, here it looks really nice, no? Spread out on the table. In 3D, this looks like a cardboard tube. They are really wrapped around each other, these beta sheets. Okay? So, what you're looking at here is the structure down the axis of the tube formed by the beta strands that are all curved up. And here you can see 2 disulfide bonds on the top and 2 disulfide bonds on the bottom. So we have like 2 layers. Now, it would seem like then you can open this structure this way and this way. You can't. Okay? You have to really look at this in 3D and rotate it a lot to find out that the connectivity of these disulfide bonds makes full denaturation of this thing geometrically impossible without breaking the disulfide bonds. At least as far as I've seen. Okay. So, we have the native state. And we know that that guy is completely soluble. Okay? Then you place it near to a hydrophilic layer, say an air-water interface, and it stretches out a part of the structure. And then this allows it to interact with the surface, and then this high-energy state is stabilized. You get enough of these guys side by side, and you make an amyloid fiber. This is a friendly amyloid. Amyloid fibers have really nasty reputations, Alzheimer's, Parkinson, and the like. This is a good one because it protects the fungus. Okay? So here you can see the rodlets, which in principle are formed like this, but we do not know. This is a guess. Okay? And then this is how a spore looks when it's covered in rodlets. The problem. Okay. This is not a hydrophobin. Okay? This is an example of a really bad amyloid. Okay? But what I want to show you with this figure is that we know where to start. We know what the native state of these amyloid promoting proteins look like, but we know nothing of the pathway or what the end product looks like until we can characterize a fiber at a high resolution. So this is an antibody domain, a variable domain from an antibody. Okay? That is a native structure. That's how it looks like when it is part of an antibody circulating in your blood. This is the good guy. In this, this is Dr. Jekyll, okay? Now, when you get enough of these guys in circulation, by an unknown mechanism that may or not involve sojourn in a lysosome where the pH is low, there is a partial denaturation that ends up in the wholesale denaturation of the protein, so that instead of being a beta sandwich, it becomes a flat pancake. And then these stack one on top of the other to infinity and beyond, and that is an amyloid fiber found in the heart of a patient. Okay? Before we got that structure in 2019, all the models that were made for the fibers made with this guy were very native-centered. We were all thinking that whatever the intermediate was very close to the native state, because this is already full with beta sheets, so beautiful, huh? And we want a structure, the amyloid fibers are rich in beta sheets, so we were done. So what I want you to show you with this case is that really knowing the native states gives you absolutely no information on how this is going to look in the fiber. To make matters even worse, we know that amyloid formation is under kinetic control, not thermodynamic control. That means that depending on how you coax your protein to unfold, you are likely to get a different structure. Okay? So with the hydrophobins, for example, they use as a substrate the cell wall of the fungus. That is very poorly defined at a molecular level. For once, I do not know what is the exact conformation of everything that is on that layer, and if it changes when the fungus is stressed or not, or depending on the medium where it grew. Okay? So all of these conditions might lead to different protein conformations and therefore to different fiber conformations. So we're not going to attempt going all the way over here. We're going to look at what happens near here. That said, the main idea of our work is that the formation of rodlets requires a population of an N-star structure, where N is native and star is an excited state. Okay? It is different from the native state, but it has all the disulfide bonds. Why? Because we know experimentally that you need all the disulfide bonds for this creature to make the rodlets. If you reduce them, no rodlets. So that's the only information we have. Okay? Now, in order for the apparition of N-star, we need specific enhanced flexibility at adequate sites, wherever this protein has aggregation-prone sequences. So the protein can move around a lot, but if it always hides the sequences that allow it to aggregate sideways, then these motions are useless. No? Okay, so finally, the difference between, well, among the hydrophobins that we study is the environment in which N star is more accessible. So say you have a family of proteins and you submit all of them to the same stress. And then you ask them, how willing are you to unfold in the right way? The one that makes it easy, that makes the jump easier, that is your best hydrophobin. Okay? So these are our guys. We have 4 fungi, Neurospora crassa and Aspergillus nidulans. These are mesophiles, so they grow in the absence of salt. Actually, they colonize bread. If you forget bread, that's what you get. The orange one, that's Neurospora. Now, Aspergillus idowi is from the same family as Aspergillus, well, it's the same genus. This guy is halotolerant. It can grow very nicely up to 1 molar sodium chloride. It was isolated from sugarcane bagasse. So when you crush sugarcane to get syrup, the cellulose that's left and all the junk that you cannot use, well, that was used to, and also in the presence of a lot of salt, to see what would grow there. And this guy grew. Okay? It has also been found infecting corals. So it likes to live in the sea. For reference, the sodium chloride concentration of seawater is 0.5 molar. So this is above what the sea likes. And then our champion is Balemia ichthyophaga. These 3 are ascomycetes. This is a basidiomycete. And this is a strict halophile. This guy only grows in a lot of salt, where a lot is 5 molar. This is 1 blink away from precipitation. Okay? Now, what is here on the side is the name of the proteins that we're using from each fungus. So we have 2 mesophiles, and then we have 3 halotolerant and 3 strict halophiles. This is a busy slide, but I'll walk you through the important part. So we have the organism, the name of the hydrophobin, which is the condensed sequence characteristics. That is, what is the number of residues between particular cysteines, also the net charge of the protein, and what is the sodium chloride at which this protein is expressed. So where it says no, it means that it is nominal salt, say 50 millimolar. No? Now, what do you do when you get a table like this? If you like bioinformatics, you start seeing what you can learn from the sequence. So which could be the usual suspects? Of course, the net charge. No? I told you that when you want to live in a lot of salt, you have to be net negative. So you look for the most negative, it's this guy, and the least negative is this guy. And they both grow, they are both expressed under no salt conditions. So there goes that hypothesis. That was not it. And you say, okay, maybe the length of whatever is between the disulfide pairs. No? So you just have to look at each of the columns in between cysteines. And believe me, there is no correlation. Okay, whatever is in blue is either the largest number or the smallest number for that column. And if you then go look at salt concentration, no message there. That means that bioinformatics will only get you that far, and it gets you nowhere. So you have to go to the next level of information, which is 3D information. Okay? So that's where we enter. That's our expertise. So what we did is molecular dynamics with increasing salt concentrations. In order to do that, you have to start with the structures of the proteins. There's only the reported structure for EAS, that's a Neurospora crassa one. But for all the other 7, we had to build homology models. By the way, artificial intelligence fails miserably for these proteins, and that has to do with the 4 disulfide bonds that the proteins have. AlphaFold gave me probably the most nonsensical answer I have ever seen reported in Twitter. And trust me, there's been a few. No? And also Robeta, in its artificial intelligence version, made a disaster. Okay? So we did the regular homology modeling with fire. That one worked like wonders. Once you have the protein structure, you get it into a simulation box with the desired sodium concentration, and it looks like this. We follow the minimum image convention, so the protein is officially at infinite dilution. That's the protein. Of course, salt, no. Salt is at the concentration that we want. We learned the hard way that when you add a lot of salt, there is electrostriction, which means that when you're equilibrating your simulation box, it shrinks, and then all of a sudden, your protein is outside the water box. So we had to rerun. And also, we found the hard way last week that these proteins are very talkative. They unfold very easily. We have many events of local denaturation. So again, we made the boxes assuming that the protein would do nothing, and it really splayed out, and it also ended up outside the box. So that's why we're repeating 1/4 of our runs. We're running for at least 1 microsecond for each protein at each salt concentration. So this is a 40 millisecond project. Okay, and 40 microsecond project. We wish it was millisecond. And then we analyze the runs for properties that are related to all the our checklist of things that could happen to the proteins. So we have sanity checks to make sure that the proteins are okay, and then things that have to do with structural properties and dynamic properties, both at very short time intervals, like 100 picoseconds, and also at the whole run level. Okay? So here we go. First question. Is 1 microsecond enough? The short answer is, of course, no. Okay? But why do we dare to present results? Well, I'm showing you a plot of the radius of duration versus time for isoform 2 of Balemia ictiophaga. Doesn't matter, one of the many proteins that we simulated at 2 different salt concentrations. And what I want you to see is that we have many changes. And so the protein starts in a compact fashion, then it yawns, stays there, comes back, yawns a little bit more. So it seems that we're getting repeat events. So this looks like it's getting close to being equilibrated. Also, if you look at the run at 2 molar, then we even have more events. So here, Marco chose a few points from the blue simulation, and you can see here the starting structure. So this blue loop is closely opposed to the protein, and then it moves out, it wiggles, and then it moves back. Okay? So at least we get them to go and come back. So normally, when you have a run of 1 microsecond, that means that you can be decently confident of things that happen on the 100 of nanosecond time scale. That is roughly the thumb rule. And it seems that local denaturation events for these proteins fall exactly in that time scale. So, first real science question, not methodological, but what we really want to find out is, are these proteins sensitive to sodium chloride? Okay, so we looked at how conformationally diverse is the ensemble of structures that we generated by clustering. This is alpha carbon clustering with a cutoff of 2 angstrom. So it's a standard practice for folded proteins of that size. Okay, so this table shows 3 proteins, the salt concentration at which they are expressed, and then for each salt concentration, how many different clusters were found with this cutoff of 2 angstrom. okay? And then over here, we have the net charge of the protein, just in case we could see any correlations, okay? So what I want you to see is that at very low salt concentration, this guy and this guy are very talkative. They change their conformations a lot, so we see many different clusters. More so for isoform 1 than 4, okay? And as we increase salt concentration, the proteins become more boring. They don't want to explore that much. This guy is weird. It was never diverse, okay? And of course, when I looked at the numbers, and again, I suspected a bug, is that what on earth did we not see? What did we not anticipate? What happened? Turns out it's a sequence feature. So it has to do with this net charge. Of course, just looking at the number gives you nothing. It matters how the charges are spread out on the surface of the molecule. So this guy has almost perfectly balanced charges on the surface, and they are scattered around very nicely, so you have no accumulation of net positive or net negative charges on the surface. On the other hand, this guy has, in particular, one loop that is loaded with negative residues. So that's why it goes bananas at low salt concentration. Okay? So we still have to analyze this more carefully, but at least now I think that this guy was acquired by horizontal transfer. So I asked Rayo and Jordanis to look at the transcript that they got from the fungus to check their codon, the codon usage for that gene. If it belongs to, if it has been with the fungus for a long time, the codon usage will be exactly the same as for the other isoforms, and if not, then it looks like it was acquired from a friend in the recent past. Okay. By the way, this is the signature that we would expect to find for a protein at 300 Kelvin, which is the simulation temperature that we used. So this is what a salt-insensitive protein looks like at 300 Kelvin. The issue is that these guys are sensitive to salt. This one isn't. Now, that was for the whole protein. What can we learn per residue? So here what I'm showing you is the root mean square fluctuations for each residue at the different salt concentrations. And I'm just showing you a really talkative one and the one that is very silent. And you can see from the scale of the y-axis is the same, so that it is easy to compare. So you can see that this guy moves around a lot, and this guy doesn't move around most of the time. Okay? Now, these fluctuations are calculated in time intervals of 100 picoseconds because there are neutron scattering experiments that measure heavy atom fluctuations. Actually, that's not true. They measure proton fluctuations in proteins experimentally. And the neutron beam can be tuned to look at precisely this time frame. And it's important because then you can guarantee that it is harmonic and not diffusive. So this is really information on the curvature of the well where the protein is sitting. Okay? So qualitatively, they look different. Now what we did is to average these fluctuations from the first cysteine all the way to the last. And when we do that, what we see is that for this guy, at no salt, it fluctuates much more than at high salt, almost twice as much. This guy couldn't care less, and this one cares a little bit more, but not much more. Okay? This was for the whole interval between the first and the last cysteine. We're not looking again at the N-terminus so far. Okay? Now, there are 3 large loops in this protein, and they are color-coded. So we have this red guy, which is region 2, the blue guy, which is region 1, and the green one, which is region 3. That's because of the order in which they appear when you go from N-terminus to C-terminus, okay? So, what happens when we do the averages, but for each region? Again, what we see is that SIH1 is salt sensitive, and in particular, that has to do with this red loop over here. And in a spoiler alert, that's the loop that is filled with negative charges. So it makes a lot of sense, okay? The others, it doesn't matter which loop you look at, they don't seem to respond too much to salt. This also shows us that the protein is dynamically heterogeneous. So whenever you're analyzing simulations and you're looking at RMSF, to root mean square fluctuations, plot them. Separate them into different regions. Learn more about your protein. This has nice information. Now, the model that we have in mind is like one of these tea flowers. I don't know if you've seen them. They sell them in Chinese stores, Chinese tea stores. So it's a flower. It's all crumpled up, and when you put it in hot water, it blooms. Okay? So we're thinking that this protein does the same thing, and we have 3 loops. So the question is there a specific order in which they should be opened, or they don't really care? So in an analogy with a box, say you want to open this box, first you have to open the outer lids, and then you can open the ones in the middle. So, to find out if there is a favorite order, what we did is to calculate these conformational landscapes where we have on each axis one of the loops. So what you're looking at, each point on this histogram corresponds to the values for the radius of gyration of region 1, which is the blue guy, region 2, which is the red guy, or region 3, which is the green guy, for all the snapshots in the simulation. So it's 1 microsecond, okay? And this is only at the lowest salt concentration, and I'm showing that because that's where it's clearer, okay? It's easier to see. So, if we look at the effect of region 1 on region 2 and vice versa, we see that there is no effect. They really couldn't care what they're doing. And if you look at the structure, it makes sense. Region 2 is over here, and region 1 is over here, so they don't talk to each other. On the other hand, if you look at this plot, what we see is that when region 3 is closed up, then region 1 cannot open. You have to first unfold region 3, and then this guy can open up. Okay? Now we have to understand exactly why, so we have to extract photographs from here, and then photographs from here, and then we can figure out what was lost and what was gained in each scenario. But it seems that, at least, region 2 is fairly independent, while for rod A, which is another of the mesophiles, region 3 could be holding region 1 in place. Now, again, the protein can yawn as much as it wants, but if it's not exposing amyloidogenic zones, I couldn't care less. I'm looking for N star, and N star is this structure that can serve as a seed for rodlet formation. That's the idea. So using Waltz as a predictor for amyloidogenic zones, we found how many zones were present in each protein, and what I'm showing you here is the original structure, and this is SIH4 with region 1 almost completely stretched out, and what is seen here in purple is an amyloidogenic zone. So in this case, this would be a candidate for N star because it is exposing very nicely a very long amyloidogenic zone. Now, most of the time when one performs molecular dynamic simulations, one goes for the mean. One looks for whatever the bulk is doing. Now we're going to use the data to look for miracles. We want to find high energy states. So those are clearly not going to be good, adequately described by the mean and the standard deviation of any property that we calculate. So what do? In order to catch n star, first we take the complete interval of values for a particular property at all salt concentrations. So we're asking the protein, what could you do in the 5 microseconds that I looked at you? Were you very quietly sitting down, or were you stretching out nicely? What on earth were you doing? Okay? That gives us an interval. We break that into 10 subsets, and we are going to take the 10th percentile, so the highest values for both radius of duration and exposed surface area. Okay? And then we go back and we count for the number of events that happen at each salt concentration for each protein. Okay? So let's look at 2 examples. So if we look at the radius of duration, again, this is just SIH1. This is the one that is very, very diverse in its conformations. Here we have what each of the regions did at different salt concentrations. This number gives me the number of snapshots that I find in my complete set, which is of 1 million. Okay? So we're talking about something that happens 1 in 1,000. Okay? Also, I want you to see that when the salt concentration is very high, there are no events, and that is in agreement with the fact that high salt makes the protein compact. Okay? Okay, so that is radius of duration, and now what does that do for me regarding amyloid zone exposure? These calculations are more expensive, so in order to have a quick and dirty idea of what was going on, we calculated that with the skip of 1,000, so here we have only 1,000 points. Okay? That's why these numbers are smaller. Not because the events are fewer, but because our data set is smaller. So we have, SIH1 has 8 amyloidogenic zones, and they are spread along its structure in the amino terminus, in region 1, region 2, and region 3. And I want you to see that at low salt, there are a lot of events where an amyloidogenic zone is exposed. Now, if we do the same analysis for SIH2, I want you to remember that this guy was very timid. It barely moved. Okay? So that means that its intervals are really tiny. That's why we get a lot of events. But the amplitude of those things are really small. So for this guy, I want to perform a high temperature simulation to see what it's really capable of doing. Okay? Anyways, with this caveat in mind, turns out that most of the time this guy does not expose anything. So we propose, based on this analysis, that SIH1 is a good hydrophobin because it yawns very frequently, and in those yawns, it exposes sticky zones. On the other hand, SIH2 is a really lousy hydrophobin because it is timid, and on top of that, it has very few amyloidogenic zones. Curiously enough, if you compare the levels of expression of these proteins, SIH2 is made at larger amounts than SIH1. And my guess is that that has to do with the fact that this guy is a really bad customer. It doesn't want to perform its function, so you have to make a lot of the protein to guarantee that you have enough concentration of N star to start the process. On the other hand, these guys, with a few, you have enough to nucleate the formation of the rodlets. The nice thing is that these can be tested experimentally. Rayo and Jordanis have the clones for these proteins, and they already have the expression system. And they can measure the contact angle of drops of water with a functionalized surface with these things. So my guess is that with really tiny concentrations of SiH1 compared to SiH2, you get the same angle of contact. So that's the other thing, no? When you make simulations, you have to predict something. Whether it's true or not, that's another story, but you have to predict something. Touching with reality, okay? We know really nothing high resolution about how the intermediate looks like. But Pham and collaborators in 2017 carried out circular dichroism experiments on hydrophobins when they were subjected to high steering, so that generates lots of air-water interfaces, and also when they gave them quartz surfaces so that they could deposit. So one of the things that they found is a signal in circular dichroism in the region around 300, and that is either aromatics that are changing their conformations or the isomerization of disulfide bonds. So we calculated the dihedral angle of the 3 disulfide bonds that are flanking regions 1, 2, and 3. And out of those 3 disulfides, the only one that isomerizes is C2C5. So what I'm showing you here is, again, a 3-dimensional histogram. So okay, rotating. This is the dihedral angle of the disulfide bond. It started here, and it isomerizes to this conformation. And radially, what we have is the radius of gyration of region 2. Okay? So you can also see that when it is in this conformer, it can acquire larger radii of gyration than when it is down here. The sad part is that these motions are not correlated, because the best thing that could have happened is, first, that this particular dihedral was jumping from one state to the other throughout the whole simulation. That's not true. It started here, it jumped once, it stayed put here. But the radius of gyration is changing all the time. So these things are not mechanically coupled, okay? Now, this is how the protein looks in the beginning, and this is the loop in question. This is the disulfide. This is how it looks like in its largest yawn, when it stretches the most. So this is, again, one possible end star for this protein. And you can see that this thing that was nicely folded against the protein now is completely extended. This is the disulfide that isomerized. This long blue thing is region 1. It used to be an alpha helix that is long gone. And this green thing also had an alpha helix. It's also gone. This gray thing is the amino terminus. And it's also linked with a disulfide bond. We will look at that one too, because it might be important for loosening the amino terminus. So finally, these are my findings. I don't know how I'm doing time-wise, but it doesn't matter. I'm not going to read them. If you want to, you can look at them. I've been telling you all this along the talk. Basically, the first block is that bioinformatics is not enough. The second block is that, okay, 1 microsecond is decent, not heroic or anything like that, but it's okay. And then, that in general, salt tightens up the protein. Okay? Also that some proteins and their loops are more sensitive than others. So that makes us think that having different paralogs is important for the fungus. Balemia has a genome that is this size. It is really small, but it has 35 paralogs for hydrophobins. So they must be important for something, okay? We found at least a couple of end stars, which is what we were searching for. And that means that we can see how they are formed, which contacts are broken, how to make that more efficient. Because again, having the clone for the proteins means that we Rayo and Jordanis can perform site-directed mutagenesis wherever they please. And they are asking us for suggestions, and we can provide them. So that's nice. And also, we found that a sulfide bond that isomerizes, and this is consistent with a circular dichroism signature. So that is our only anchor with experiments so far. But we're looking for more. Finally, I want to thank you for your patience at this pre-lunch hour. All the simulations and calculations that I showed you were performed by Marco Ramirez back there. It's not his PhD thesis. This is a side project. And all the biology comes from Jordanis and from Maria del Rayo. And hopefully, we'll get to the point where we can connect. Funding comes from Rayo's Fronteras project. We have supercomputing in-house. That comes from other seed projects in prehistory. And of course, we parasite almost all supercomputing centers in the country. We're only missing Guadalajara, but we're working on that. So thank you very much.
Okay, we have time for 1, 2 questions.
So the rodlets integrate all types of the hydrophobins in a mixture?
That I don't know. That would be okay, I have to read up more on that. But I know that experimentally you can make rodlets from only one type.
And the second question is, in some protein aggregation problems, the aggregation is catalyzed by the misfolded protein. Is there any indication that hydrophobins self-catalyze their rearrangement?
Prion-like? I don't know. But it might not be necessary because these loops appear to be held by pins. So you just look at them the wrong way and they unfold. So I wouldn't see it as necessary. Again, I don't imagine the protein becoming completely flat. It will look more like a head with tentacles because the nucleus of disulfide bonds doesn't have the right topology to allow for complete extension of the beta sheet. No? But still. Thank you.
All right, any other question? So we thank again Professor